So what we're going to cover tonight is a continuation of we've been talking about, for example, the notion of archetypes that Jung came up with that would be key to shaping people's lives. And the archetypes show up in all kinds of ways, for example, in fairy tales. But it's part of a larger theme, which we've dealt with many times before. We've always tried to deal with the parable of the blind man and the elephant, and where the blind men are all blind, and the first one goes up and grabs the elephant's tusk, and the next one grabs the elephant's trunk, and the next one grabs the elephant's ear, and the next one grabs the elephant's tail, and then they grab the side of the elephant and the leg of the elephant. And so everybody now knows what the elephant is, and they all defend that they're blind, but they all know they've seen the elephant. It's there, and they have these fights over what it is, and they all miss the point that there's an elephant sitting there that they're all dealing with. So the idea here is we start with something like Plato, who undoubtedly got this from the Egyptians, about the notion of forms or shadows. And Jung, for example, picked up on this in his ideas of archetypes and so forth. But it shows up in many other ways. We touched on it in mathematics, where there was a mathematician named Langlands, and he said, oh, there's linkages between all these very diverse areas of mathematics, in particular, in between number theory, dealing with typically looking at numbers and trying to find prime numbers, and harmonic analysis. And you'd ask yourself, how could there be any linkage whatsoever between, prime numbers and harmonic analysis? But he proposed this, and now there's actually linkages to geometry that have appeared, and also just it appears that Jung was dealing with the notion of the integers as being archetypes, 1, 2, 3, 4, 5, as being archetypes. There was something that he saw there, which we'll come back to in the next class, and so forth. And so in mathematics, you have this drive for these linkages, or the trying to see what it is, and called this meta mathematics because it's above, it's the meta, it's above what we would call mathematics. It's the bridges that interconnect all these different things.
Principles.
So forth. So tonight we touched on this last time looking at forms, but these are forms that are implicit in your DNA and RNA to create the different parts of your body, your eyes, your fingers, your bones that up that are there encoded in this DNA RNA, which is not obvious. Where is this encoded? And so Michael Levin has dealt with picking up on the work of Darcy Wentworth Thompson in this regard, who felt that there was much more to biology with regard to these shapes or forms or archetypes or shadows, if you will. And so that's what we're going to deal with tonight. So, Marjorie, what would you like to say?
Well, I'm thinking about how Jung's thinking about the archetypes and synchronicity evolved over the years. And I don't think he understood what he thought in the end. I don't think he thought that in the beginning. And I certainly didn't. And when I was young, say 30, and first studying it, I thought he was contradicting himself. It didn't occur to me that I was seeing things over a period of, 30 to 50 years of writing about it, and that his own thinking changed, vastly over the years. And for example, I think in the end, he really believed that synchronicity is causal, but in a different way from the way we normally think about it. It's just kind of a simplistic 1, 2, A, B, um, that it's more subtle and complex, and that things like we're talking about when strands of DNA are, found in their natural state in the body, they're all folded up and compressed. So different components are next to each other because they're folded up and compressed. And we might think of them as in their stretched out state, which is about 6 feet long, right?
Right, for humans.
The length of average human male body,
Hmm.
and that when they're stacked, they're right next to each other. And I think a lot of things in life are like that. And what we think of as, particle physics and so on, there's a lot more of that involved than we ever dreamed of. Um, and, one way to look at archetypes is what Plato was talking about in his ideal forms. I do think that was one. And Jung ended up thinking that whether it's a mythological archetype, he felt that dreams are individual myths and myths are collective dreams. And that's one way we can see the archetypes, and that they have their own existence. They are pre-existent with us or without us. And in a way, they're living us, they're dreaming us. And we are expressions of them rather than the other way around, which is certainly how I looked at it in the first, decade or so of my study and experience, and seeing the way that they play out in our art. I don't think it was conscious design by the people creating and morphing and passing along the fairy tales and the myths. I think they were bubbling up of their own accord. And I think we have, a lot of examples of that in history and um, in our social history and our religious history in particular. I think those show the archetypes kind of percolating over a period of time and a huge group of people. One person didn't write the story of Christianity. That evolved over many years before the birth of the person we think of as Jesus Christ. Long before that, it was percolating, and since then, I think it's continued evolving and morphing, and that's what the theme that's happening. Our consciousness, as well as our brains and everything else, uh, uh, I would say the same with the ideas of Buddhism, and that the Buddha was another archetype, really around the same time period. That was another example of the Piscean symbolism of the age of Pisces. Whether people know or believe it was the age of Pisces at that time, those symbols were very prevalent in what was coming out in human expression. So they affect us, whether it's conscious or not, whether we believe it or not. It's really as if they're dreaming us and our lives are fitting into it. For the first time in a long time, I just knew that Mercury was retrograde on the second day from, a few patterns that I was just observing in our microcosmic life. And I've known people who didn't really know astrology, but they would know within a couple of days. And I don't usually. I usually have to look it up. I looked it up to confirm it, but I suspected it beforehand. So, you know, I, that's a very abstract kind of thing, but it's also very concrete and very universal. You see images from just about every culture. They might have a different name for Mercury, and the story of the god and the adventures, which these people who taught at MIT and wrote this book,
Hamlet's Mill.
Hamlet's Mill, thank you, wrote about, that the myths seem to be mnemonic devices for remembering these cycles, which are very regular and very consistent. So they had to be observed over long periods of time and passed on in various ways over long periods of time. And the theory of this pair of scholars was that that's what the myths were really for, was mnemonic devices to remember and pass along the regular cycles and to represent it. And that's really where the story of Hamlet from Shakespeare came from. The much older story. It's an old Danish myth that, probably ultimately came from Sumeria or someplace over there. And it's really about the cycle of Saturn and Jupiter, which you can show as a geometric diagram, and you can also have stories about their cycles of overtaking one another. So I think Michael Levin has a really fascinating handle on it, and how it this idea of archetypes, or what's another word?
Forms.
The platonic forms. So the triangle and the square and that we all kind of recognize them. They're universal in some way. And as he says, they're idealized. They don't exist that way in nature. But we all agree on, the angles, the degrees, the math of it. And so they're another example of an archetype.
If I wanted to pick up on what Ron Cowan observed. He under deep meditation,
The brother of Bruce, who's
Bruce's brother, that there was information blocks that coded the discrete compartments of space. The space is actually a foam that in a very micro level that's coded in the insides of each space compartment with blocks of information that is constantly scanning the world around it, and then has algorithms. The algorithms are the archetypes. Okay, they store what they observe, but they're able to control the world around them. So the archetypes, through the algorithms, actually control the world that we live in, the different forces that we see all around us. And what's amazing is this occurs on all levels. So Ron saw it occurring right down at the very bottom of space itself. But as we see, the very simple algorithms can lead to incredible complex behaviors that then evolve, that iterate on themselves. And that's what we observe in daily life, is all of these things happening. And so above as below is the message to take away from all this. So I think with that, let's try and get into what Michael Levin has to say. And we're going to go to his video here. And give a yell, Bruce, if you can hear this. I'm going to go here. I'm going to go, we got captions on. I'm just going to speed it up a little bit. There we go. And
the hardware does not define you. I get lots of emails from people who say, I've read your papers, I understand I'm a collective intelligence.
Yeah, I can hear.
okay.
What do I do now? I just learned that I'm full of cogs and gears, therefore I'm not what I thought I was. And I think this is a really unfortunate way to think. The bottom line is you are still the amazing integrated being with potential and a responsibility to do things. I think we are actually a collection of interacting perspectives and interacting consciousnesses.
Professor Michael Levin, welcome. What's one of the biggest myths in biology that you, with your research, with your lab, is helping overturn?
Yeah. Hey, Kurt. Great to see you again. I think that one of the biggest myths in biology is that the best explanations come at the level of molecules. So this biochemistry, what they call molecular mechanism, is usually currently taken to be the kind of the gold standard of what we're looking for in terms of explanations. And I think in some cases that's valuable. That's that has the most value. But in most cases, I think it's not the right level. And Dennis Noble has a really interesting set of papers and talks on biological relativity. So this idea that different levels of explanation, especially in biology, provide the most bang for the buck. And I think in terms of looking forward as to what do these explanations let you do as a next step, I think we really need to go beyond the molecular level for many of these things.
What's the difference between weak emergence and strong emergence?
I think that emergence in general is basically just a measure of surprise for the observer. So I think a phenomenon is considered emergent by us if whatever it's doing was something that we didn't anticipate. And so it's relative. I don't think emergence is an absolute thing that either is there or isn't, or is weak or strong. It's just a measure of the amount of surprise. How much extra is the system doing that you, knowing the rules and the various properties of its parts, didn't see coming. That's emergence, I think.
Would you say then that biology has physics fundamentally? So it goes biology, chemistry, physics?
I mean, people have certainly made that kind of ladder. I'm not sure what we can do with that. I think that it's more like these different levels have their own types of concepts that are useful for understanding what's going on, and they have their own autonomy. That's really important. That I think it's there's a lot of utility in considering how some of these higher levels are autonomous and they do things that the lower levels don't do, and that gives us power. That gives us experimental power.
And would you say that there's something that the lower levels don't do in principle or in practice? So for instance, is there something that a cell is doing that in principle can't be derived from the underlying chemistry, which in principle can't be derived from the underlying physics?
Yeah, it really depends on what you mean by derive. But for example, there are certainly aspects of, let's say, cognitive functions that we normally associate with fairly complex creatures, brains, and so on, that we are now seeing in very simple basal media. So even something as simple as a gene regulatory network can have 6 different kinds of learning and memory, for example, Pavlovian conditioning. And so there, what you would see is on the one hand, you say, okay, well, look, here's a simple bit of chemistry that's actually doing these complex things that we normally associate with behavior science and with investigating cognitive systems. You can always tell a chemistry story about any effect. In fact, you could also tell a physics story as well. If you look under the hood, all you ever find is chemistry and physics, right? But the more interesting thing about that story is that you actually can use paradigms from behavioral science, such as learning and conditioning and communication and active inference and things like that. And that lets you do way more than if you were to restrict yourself to an understanding at the level of mechanism.
What's super interesting there is that you said you could always tell a chemistry story or you could always tell a physics story to explain some biological process, but you can't always tell a biological story to something that's physics related. So what people would consider more fundamental, or what gives rise to the rest in terms of ontology, would be, well, can you tell a story in terms of so-and-so? So can you tell a story in terms of biology, of physics? Can you tell a story of, well, you can tell a story of architecture for these buildings behind us, maybe even of biology, because people comprise the people who made this, and you could tell a mathematical story, but it would be difficult to make the case that you could tell an architectural story in the way that we understand architecture, not a metaphor, about mathematics. Do you agree with that, or do you disagree with that?
I think that's somewhat true, although it's not as true as we think. So oftentimes you actually can tell an interesting story cashed out in the terms of behavioral science. For example, active inference, for example, various learning modalities of objects that are really thought to be the domain of physics and chemistry. So I think there's more of that than we tend to understand as of now. But it is also the case that the fact that you can tell a physics story doesn't mean that there's that much value in it necessarily. So for example, imagine that there's a chess game going on. So you could absolutely tell the story of that chess game in terms of particle movements or maybe quantum foam, or I don't know whatever the bottom level is. So you can do that. How much does that help you in playing the next game of chess? Almost not at all, right? It's if your goal is to understand what happened at the physical level, yes, you can tell a story about all the atoms that were pushing and pulling and all of that. But if your goal is to understand the large scale rules of the game of chess, the principles, and more importantly, to play the next game of chess better than what you just witnessed, that story is almost completely useless to you. And this abounds in biology, where you can use chemistry and physics to tell a story looking backwards about what just happened and why. But in terms of understanding what it means and developing that into a new research direction, new biomedicine, new discoveries, it often means that you actually have to tell a much larger scale story about memories, about goals, about preferences, and these other kinds of concepts.
What would be the analogy here for, so the rules of chess? What are you trying to understand? The rules of what? Biology, developmental biology, cellular biology?
Well, the thing that ties all of our work together, I mean, we do a lot of different things. We do cancer biology, we do developmental biology, we do regeneration, aging, not to mention AI and some other things. What ties it all together is really an effort to understand embodied mind. So the center focus of all my work is really to understand cognition broadly in very unconventional, diverse embodiments. And in biology, what we try to understand is how life and mind intersect, and what are the features that allow biological systems to have preferences, to have goals, to have intelligence, to have problem solving, clever problem solving in different spaces.
Okay, great. Because there are a variety of different themes that your work touches on. So regeneration, cancer research, basal cognition, cross-embryo morphogenetic assistance, which is from a podcast that we did a few months ago and that will be on screen, link in the description. There's xenobots, anthrobots. Please talk about what ties that all together.
Yeah. What ties it all together is the effort to understand intelligence. All of those things are really different ways that intelligence is embodied in the physical world. And so, for example, when we made xenobots and anthrobots, our goal with all of these kinds of synthetic constructs is to understand where the goals of novel systems come from. So typically, when you have a natural plant or animal and it's doing various things, not only the structure and the behavior of that organism, but also whatever goals it may have, we typically think of are driven by an evolutionary past, right? So these are set by the evolutionary history, by adaptation to the environment. So, for eons, everything that wasn't quite good at pursuing those goals died out, and then this is what you have. That's the standard story. So the reason that we make these synthetic constructs is to ask, okay, well, for creatures that never existed before, that do not have an evolutionary history of selection, where do their goals come from? So that's just part of that research program, to understand where do the properties of novel cognitive systems come from that do not have a long history of selection as that system. Everything else is that we do is an extension of our search for intelligence. So basically cancer, right? So the way we think about cancer is as the size of the cognitive light cone of living or of cognitive systems. So what I mean by that is every agent can be defined by the size of the largest goal it can pursue. So that's the cognitive light cone in space and time. What are the biggest goals that it can pursue? So if you think about individual cells, they have really sort of humble single cell scale goals. They have metabolic goals. They have proliferative goals and things like that. They handle the situation on a very small single cell kind of scale. But during embryonic development and during evolution in general, policies for interaction between these cells were developed that allow them to scale the cognitive light cone. So groups of cells, for example, the groups of cells that are involved in making a salamander limb, they have this incredibly grandiose goal in a different space. Instead of metabolic space, they are building something in anatomical space. So they have a particular path that they want to take in the space of possible anatomical shapes. So when a salamander loses their limb, the cells can tell that they've been pulled away from the correct region of anatomical space. They work really hard. They rebuild. They take that journey again. They rebuild that limb, and then they stop. And they stop because they know they've gotten to where they need to go. That whole thing is a navigational task, right? And there's some degree of intelligent problem solving that they can use in taking that task. So that amazing scale up of the cognitive light cone that from what the light cone measures, what do you care about? So if you're a bacterium, maybe you care about the local sugar concentration and a couple of other things, but that's basically it. If you're a set of salamander cells, what you really care about is your position in anatomical space. Do we have the right number of fingers? Is everything the right size, and so on? And so understanding that scaling of cognition, the scaling of goals, how does a collective intelligence work that takes the little tiny, very competent tiny components that have little tiny light cones, and how do they come together to form a very large cognitive light cone that actually projects into a different space, anatomical space now instead of metabolic space. So that's fundamentally a question of intelligence. And the other thing about it is that this kind of thinking, so this is really important, that this kind of thinking is not just sort of philosophical embellishment, because it leads directly to biomedical research programs. If you have this kind of idea, what you can ask is, well, let's see, then if cancer is that type of phenomenon, it's a breakdown of that scaling, and that basically individual cells just shrink their light cone back down to the level of a primitive microbial cell as they were once, and that boundary between self and world now shrinks, whereas before the boundary was that whole limb. Now the boundary is just every cell is the self. So from that perspective, they're not more selfish, they just have smaller selves. So that's really important, because a lot of work in cancer models cancer cell behavior from a perspective of game theory, like they're less cooperative and they're more selfish. Actually, I'm not sure that's true at all. I think they just have smaller cells. And so
just a moment. So you would say that every organism is equally as selfish, it just depends on their concept of self?
Yeah, I think all agents are, one of the things agents do is operate. It's not the only thing they do, but one of the things they do is they operate in their self-interest, but the size of that self can grow and shrink. So individual cells, when they're tied into these large networks, it'll be using electrical cues, chemical cues, biomechanical cues, they're tied into these larger networks that partially erase their individuality. We can talk about how I think that happens, but what you end up with is a collective intelligence that has a much bigger cognitive light cone. The goals are much bigger, and then of course you can scale that up. I mean, humans have enormous cognitive light cones, and whatever's beyond that. But that's the thing. It's the size of your cognitive light cone that determines what kinds of goals you can pursue and what space you pursue them in.
So we talk about goals and we talk about intelligence, and you said something interesting, which is that life is the embodiment of intelligence or something akin to that, the embodiment of intelligence. So it's as if there's intelligence somewhere and then you pull from it and you instantiate intelligence. So is that the way that you think of it? Let me be clear about what I mean. In philosophy, there's this concept of universals. So Plato had the forms,
Yeah.
and then would say that this is almost rectangular, so it's embodying the rectangularness, which is somehow somewhere out there akin to what I imagine you meant when you said intelligence is there being embodied by this. But then there's Aristotle, which says, okay, yes, there is something akin to a form, it's just not out there. It's actually in here. The rectangularness is a property of this microphone. So I imagine this latter view is the one that most biologists would take. Not that there's intelligence out there, let's just grab it and instantiate it here. No, something has the property of intelligence. So explain what you mean when you say embodying intelligence, and also what you mean by intelligence.
Yeah. Let's see. And just a quick thing to finish real quick, the previous thought, which is that the reason I think this stuff is not these are not questions of philosophy. These are very practical questions of science because they lead to specific actionable technology. So the idea that what's going on in cancer is a shrinking of the cognitive light cone leads directly to a research program where you say, well, instead of killing these cells with toxic chemotherapies, because I believe that they're genetically irrevocably damaged, maybe what we can do is force them into better electrical networks with their neighbors. And that's exactly what we've done. And so we've had lots of success expressing strong human oncogenes in frog embryo cells, and then leaving the oncoproteins be, but instead making sure that they're connected into tight electrical networks with their neighbors, and they normalize. They make nice skin, nice muscle. They do their normal thing instead of being metastatic. And so that kind of thing, it's very important to me that all these ideas, and so we'll talk about in a minute about Platonic space and things like that. It's important to me that all of these ideas don't just remain as kind of philosophical musings, but they have to make contact with the real world, and specifically not just explaining stuff that was done before, but facilitating new advances. They have to enable new research programs that weren't possible before. That's what I think is the value of all of this kind of these deep philosophical discussions. So let's see. With respect to the definition of intelligence, so I like for practical purposes, I like William James's definition, which is some degree of the ability to reach the same goal by different means. And that presupposes, it's an interesting definition because it presupposes that there is some problem space that the agent is working on, but it does not say you have to be a brain. It doesn't say you have to be natural versus artificial. It doesn't say any of that. It's a very cybernetic definition. What it says is that you have some amount of skill in navigating that problem space to get to where you want to go, despite interventions, despite surprise, despite various barriers in your way. How much competency do you have to achieve that? And that was his definition of intelligence. Now, I will certainly agree that that doesn't capture everything that we care about in cognition. So a focus on problem solving doesn't handle play, and it doesn't handle emotions and things like that. But for the purposes of our work, I focus on the problem solving aspects of intelligence. So within that, your point about the Platonic space. So now to be clear, everything that I was saying before, I think we have very strong empirical evidence for. And so now I'm going to, in answering this question, I'm just going to talk about stuff that I'm not sure of yet, and that these are ideas we don't, I don't feel strongly that we've shown that any of this is true. But this is my opinion, and this is where our research is going now. I actually think that the Platonic view is more correct, and I know this is not how most biologists think about things. I think that in the exact same way that mathematicians that are sympathetic to the Platonic worldview, this idea that there are, in fact, in some way, there is a separate world in which various rules of number theory and various facts of mathematics and various properties of computation and things like that, right? That there's a separate space where these things live. And importantly, the idea is that, they think that we discover those things. We don't invent them or create them. We discover them, that and that they would be true still if all the facts of the physical universe were different. They would still, be true, right? I extend that idea in a couple of ways. One is I think that what exists in that Platonic space is not just rules of mathematics and things like that, which are in a certain sense low agency kind of objects because they just sort of sit there doing, they don't do much. I actually think that there's a spectrum of that, and some of the components of that Platonic space have much higher agency. I think it's a space of minds as well, and of different ways to be intelligent. And I think that just like when you make certain devices, you suddenly harness the rules of computation, rules of mathematics that are basically free lunches. There are so many things you can build that will suddenly have properties that you didn't have to bake in. You get that for free, basically, right? I think intelligence is like that. I think some of the components of that Platonic space are actually minds, and sometimes what happens when you build a particular kind of body, whether it's one that's familiar to us with a brain and so on, or really some very unfamiliar architectures, whether they be alien or synthetic or whatever. I think what you're doing is you're harnessing a pre-existing intelligence that is there in the same way that you harness various laws of mathematics and computation when you build specific devices.
Okay, well, that's super interesting. So at any moment, there's Michael Levin, but there's Michael Levin at 11 a.m., then there's Michael Levin at 11:01 a.m., and it's not clear they're the same. So there's the river, do you step in it? This is the same river. Okay, cool. there's a through line. There's something there though. Maybe it's something like you're akin to what you were an epsilon amount of time previous, and then that's the through line. So as long as that's true, then you can draw a worm of you throughout time. Are you saying that at any slice of that, there was a Michael Levin in some Michael Levin space, which is also in the minds of human space, and you're picking out points somehow? You're traversing this space of minds.
That's actually even more specific than what I was saying. I mean, that's interesting, and I think that's what I was saying, that in general, kinds of minds. So there are different. I don't know quite what to think about individual instances, but kinds of minds. You know,
because that would be an extremely large space then.
Well, I think yes, I think the space is extremely large, possibly infinite, but in fact, probably infinite. But I think what I was talking about was mainly types of different minds, types of cognitive systems. Now, for individuals, you raise a really interesting point, which is I call them selflets, which are the kind of the thin slices of experience that
you call them selflets.
Selflets,
uh-huh.
Because you have a self, and then the different slices of it are the selflets, and you can look at it as, you know, kind of like that special relativity, bread loaf, sliced into pieces, that kind of thing. So I think what's interesting about asking that question about the self and what is the through line is what's really important there is to pick a vantage point of an observer. Again, kind of akin to what happens in relativity. You have to ask from whose perspective. So one of the things about being a continuous self is that other observers can count on your behaviors and your properties staying more or less constant. So the reason that we identify, oh yes, you're the same person as you were before, is basically nobody cares that you have the same atoms or you don't, or the cells have been replaced. What you really care about is that this being, I can have the same kind of relationship with them that I had before. In other words, they're consistent. I can expect the same behaviors, the things that I think they know, they still know, and so on. And so that of course in our human lives that often breaks down because humans grow from being children to being adults. All their preferences change. The things that they remember and the things that they value change. In our own lives, we sometimes change, right? And that's a much more important change. Are you the same person, even though if all your material components remain the same, but you changed all your beliefs, all your preferences, would you still be the same person, right? So I think what we mean when we say the same is not about the matter of it all. It's about what kind of relationship we can still have, and what do I expect from you behavior-wise, and so on. And there's some really interesting, and so that's from the perspective of an external observer. Now, the latest work that I just published a couple of days ago looks at what does that mean from the perspective of the agent themselves, and this idea that you don't have access to your past, but you have access to our memory engrams, traces of past experience that were deposited in your brain and possibly in your body that future you is going to have to interpret. And so that leads to a kind of scenario where you treat your own memories as messages from your past self. The idea then that is that those memories have to be interpreted. You don't necessarily know what they mean right away because you're different. You're not the same as you were, right? Especially over long periods of time. And this comes out very starkly in organisms that change radically, like caterpillars to butterfly. So memories persist from caterpillar to butterfly, but the actual detailed memories of the caterpillar are of absolutely no use to the butterfly. It doesn't eat the same stuff. It doesn't move in the same way. It has a completely different body, completely different brain. You can't just take the same memories. And so I think what happens in biology is that it is very comfortable. In fact, it depends on the idea that the substrate will change. You will mutate your cells. Some cells will die. New cells will be born. Material goes in and out. Unlike in our computational devices, the material is not. You're not committed to the fidelity of information the way that we are in our computation. You are committed to the salience of that information. So you will need to take those memory traces and reinterpret them for whatever your future situation is. In the case of the butterfly, completely different. In the case of the adult human, somewhat different than your brain when you were a child. But even during adulthood, the context, your mental context, your environment, everything changes. And I think you don't really have an allegiance to what these memories meant in the past. You reinterpret them dynamically. And so this gives a kind of view of the self, kind of a process view of the self, that what we really are is a continuous dynamic attempt at storytelling, where what you're constantly doing is interpreting your own memories in a way that makes sense about what, a coherent story about what you are, what you believe about the outside world. And it's a constant process of self-construction. So that I think is what's going on with selves.
If it's the case then that we interpret our memories as messages from our past selves to our current selves, then can we reverse that and say that our current actions are messages to our future self?
Yeah, I think that's exactly right. I think that a lot of what we're doing now is at any given moment is behavior that is going to enable or constrain your future self. You're setting the conditions in which the environment in which your future self is going to be living, including changing yourself. Anything you undertake as a self-improvement program, or conversely, when people entertain intrusive or depressive thoughts, that changes your brain. That literally changes the way that your future self is going to be able to process information in the future. Everything you do radiates out, not only as messages and a kind of niche construction, when you're changing the environment in which you're going to live, in which everybody else is going to live. We are constantly doing that to ourselves and to others. And so that, really forces a kind of thinking about your future self as kind of akin to other people's future selves. And I think that also has important ethical implications, because once that symmetry becomes apparent, that your future self is not quite you, and also others' future selves are also not you, that suggests that the same reason you do things so that your future self will have a better life, you might want to apply that to others' future selves. Like breaking down this idea that, and I'm certainly not the first person to say this, but breaking down the idea that you are a persistent object, separate from everybody else, and sort of persisting through time, breaking that down into a set of selves, and that what you are doing now is basically for the good of a future self, I think makes you think differently about others' future selves at the same time.
Is it as simple as the larger the cognitive light cone, the better?
I want to jump in here at this point as somebody who was first introduced to a brand of Buddhism when I was 14 years old. And in Connecticut, it was an independent study, and it was what I would call Americanized Zen. It was my first text was Ram Dass's book, Be Here Now. We also read Alan Watts and so on. And I feel, for me, and maybe it's as a sun in Pisces, I'm not sure, it exacerbated already depressive tendencies that I already had. And what ended up being the most healing thing for me really surprised me. I think I needed the intellectual context of Jungian philosophy and psychology, but the thing that was the most healing was work I did on and off for about 15 years with a self-psychologist. And that doesn't mean do-it-yourself psychology. It means psychology of the self. And in this approach, the self is not seen as a thing or an object. It's seen as a process, which is what they're talking about here. And I kind of needed that re-entry into myself to start living from inside again. And I needed to integrate that with the Jungian transpersonal psychology that I was studying on my own. But I needed to ground that abstraction within this experiential, from the inside experience of self that at first really conflicted with my sort of Buddhist influence of transcending the self. And I think I think that's a good thing to be able to do, but I don't think we need to slay the ego. I think we just need to keep it in perspective. Everybody's got one, and they all have their own point of view, which is what they're talking about here. But if we can also embrace a point of view of collective selves, that, as they say, everybody's got one and they all stink. But I think a lot of the human condition at this point is that we've become like a cancer, where we have this, how do you say it, metastasis, metastasis of selves turning against the whole, because we've lost that sense of connection with all the cells being connected to, I don't know if it's a larger self or a larger non-self or the oneness. But when we start existing at the expense of every other self, existing at the expense of everyone else, it's just, constant war. Whereas it should be a harmonized effort toward a larger goal. And I just sort of love that the way they're talking about this in these very, concrete scientific terms. I think it can be shifted as a metaphor to the species as a whole. And the more sort of meditation and other work that we do, the less concerned we are just about our own individual point of view. But the fact that two selves can have this dialogue, these two men pulling this out from each other, I think is just, phenomenal. And it's just such a sense of wonder of the whole thing that we are inside behind these eyes, looking out and having these experiences that, on the one hand are extremely individual, but on the other come together in certain ways and grow. I don't know how
Yeah.
else to say it.
No,
Through
that's-
through art, through exchanging ideas, through listening to each other's music, that we're expanding that window that we have looking out. We who knows if there's really an in or an out, but in our hallucination, there's the experience of an in or an out. But I just love this idea of this cognitive light cone and expanding it. And I love this idea of repatriating these kind of what would you call them, the renegade cells that were just running amok, but they were transplanted and patriated into another organism, and they calmed down. And so they were rehabilitated rather than executed.
Mhm.
And I just love that idea. And can we do that with ourselves if we look at ourselves as, on some level, we're a macrocosm, and on another level, we're a microcosm. So, are we individual cells of the body of humanity that is kind of turning on itself right now? Um, can we turn that back to a more harmonized uh, view, if not, our understanding or experience? I think this is the kind of ideas that we need, and what Bart and I are always looking for is where science and spirituality can intersect. And I think at least on the level of metaphor, there's a lot of
Yeah.
good, borrowing and cross, exchange that we can make use of. So I think this is exciting because I think it's a way to articulate things that
Mm.
we've lost touch with.
But in a very concrete way.
Right,
So
right. It's a language if we think of
Yeah.
it in metaphoric terms.
For the organism.
Well, what does better mean? I mean, I think that certainly there are extremely successful organisms that do not have a large cognitive light cone. Having said all that, the size of an organism's cognitive light cone is not obvious. We are not good at detecting them. It's an important research program to find out what any given agent cares about because it's not easily inferable from measurements directly. You have to do a lot of experiments. So, assuming we even know what anything's cognitive light cone is, I think lots of organisms do perfectly well. But then, what's the definition of success? So, in terms of the way we think about, the way many people think about evolution, in terms of, how many copy number, like how many of you are there? That's your success. But just persistence and expansion into the world. From that perspective, I don't think you need a particularly large cognitive light cone. Bacteria do great. But from other perspectives, if we sort of ask ourselves what the point is and why do we exert all this effort to exist in the physical world and try to persist and exert effort in all the things we do in our lives, one could make an argument that a larger cognitive light cone is probably better in the sense that it allows you to generate more meaning and allows you to bring meaning to all the effort and the suffering and the joy and the hard work and everything else. From that perspective, one would want to enlarge one's cognitive light cone. And we collaborate. Well, I collaborate with a number of people in this group called CSAS, the Center for the Study of Apparent Selves, and we talk about this notion of something. For example, in Buddhism, they have this notion of a bodhisattva vow, and it's basically a commitment to enlarge one's cognitive light cone, so that over time one becomes able to have a wider area of concern or compassion. Right? The idea is you want to work on changing yourself in a way that enlarges your ability to really care about a wider set of beings, and so from that perspective, maybe you want a larger cognitive light cone.
Okay, so there are three notions here. So enlarging what you care about. Okay, then also enlarging what you find relevant. And then there's also increasing the amount of opportunities that you'll have in the future. So that's the adage, that's the business adage. Go through the door that will open as many doors as possible. And then the relevance one, I don't think is the case, because if you find too much relevant, you will also freeze because you don't know what to do. Now then there's the concern for others. So help me disembroil these three from one another.
Yeah. The relevance is I think really important. One thing I often think about is like what's the fundamental unit that exists in the world? Is it genes? Is it information? Like what is it that's really spreading through the universe and differentially reproducing and all that? I tend to think it's perspectives. I think that what's really out there as a diverse as a way to describe really diverse agents, I think one thing they all have is a commitment to a particular perspective. So perspective, in my definition, is a bundle of commitments to what am I going to measure about the outside world, what am I going to pay attention to, and how am I going to weave that information into some sort of model about what's going on, and more importantly, what I should do next. So there are many different perspectives, and as you said, it's critical that every perspective has to shut out way more stuff than it lets in, because if you and this interestingly, this gets back to your original point about the different levels of description, physics and chemistry and all that, because if you try to if you wanted to be a Laplacian demon, if you wanted to track microstates, all the particles, right? I'm going to track reality as it really is. I'm just going to watch all the particles. That's all I'm going to do. No living system can survive this way, right? Because you would be eaten before anything else happens. You would be dead. So I think that any agent that evolves under resource constraint, so which is all of life, and it remains an interesting question. What does that mean for AIs and so on? But any agent that evolves under constraints of metabolic and time resources is going to have to coarse grain. They're going to have to not be a reductionist. They're going to have to tell stories about agents that do things as a means of compression, as a way of compressing the outside world and picking up perspective. You cannot afford to try to track everything. It's impossible. So that aspect, that compression also comes back to the memory engrams issue that we were talking about, because as your past self compresses lots of diverse experiences into a compact representation, right, of a memory trace of what happened, that kind of learning is fundamentally compression. When you're compressing lots of data into a short, pithy generative rule or a memory that you're going to remember that you can use later, right? So not the exact experiences that you had, but some compressed representation. Well, the thing about compression is that the most efficient the data that's compressed really efficiently starts to look random, right? So because you've gotten rid of all the correlations and everything else, the more you compress it, the more random it looks, and you've really gotten rid of a lot of metadata that you have to. That's the point of compression. You've lost a lot of information.
Wait, why do you say that the more compressed it is, the more random it is? Why isn't it the opposite? The more random it is, the more incompressible it is.
That's true, because you've already compressed the hell out of it. That's why, right? That's why it's incompressible, because you've already compressed it as much as it can. This is the issue that, for example, the SETI people, right, the search for extraterrestrial intelligence, that they come up against, because the messages that are going to be sent by truly advanced civilizations are going to look like noise to us, because really effective compression schemes, unless you know what the so you've compressed it, unless you know what the algorithm is to reinflate it, the message itself looks like noise. It doesn't look like anything, because you've pulled out all of the correlations, all of the order that would have made sense to a more naive observer is now gone, and if you don't have the key to interpret it with, it just looks like noise. And so that means that if you sort of think about this, there's an architecture like a bow tie kind of architecture that's important here, where you take all these experiences, you compress them down into an engram, that's your memory, and then future you has to reinflate it again and figure out, okay, so what does this mean for me now, right? It's a simple rule that I inferred. Let's say you learned an associative learning task, or some kind of, you've learned this something general. You've learned to count, a number or something like this. So, so rats, I think it takes about, if I recall correctly, like 3,000 trials before they understand the number 3. That's distinct from any instance. So it's not like 3 peanuts or 3, sticks. It's like the number 3 of anything, right? So after, some thousands of trials, they get it. And so now they have this compressed rule. They don't remember the sticks or the peanuts or whatever, but they remember the actual rule to right. And so that's the engram at the center of your bow tie. Well, future you has to reinflate it, has to uncompress it and expand. Okay, but now I'm looking at 3 flowers. Is that the same or not? How do I apply my rule to this? And I think that what's important is that you can't do that deductively, because you're missing a lot of that basic information. You have to be creative when you interpret your own engrams. There's a lot of creative input that has to come in to understand what it was that you were thinking at the time. And I think that that kind of a thing. I mean, we know that recall of memories actually changes the memory, right? So in neuroscience, they know this, that there's no pure non-destructive reads of memories, that when you access your memory, you change it. And I think that's why, because the act of interpretation is not just a passive reading of what's there. It's actually a construction process of trying to recreate. So what does that mean for me? What does it mean now? And that's part of that process of the dynamic self, is trying to figure out, and obviously all this is subconscious, but trying to figure out what your own memories mean.
Yes. Okay. So you said obviously much of this is, or maybe it's obviously all of it, or much of it, I'm not sure, is subconscious. So when we say you currently are constructing an engram for the future you to then unpackage, I am not doing this, at least not at an effortful conscious level. There's an instinctual unconscious component to it. And then both to the encoding and then to the retrieval and the expansion of the engram.
Yeah.
So the person who's listening to this, they listen to these podcasts, they listen to theories of everything, in large part because they're trying to understand themselves, they're interested in science, they're interested in philosophy. You're also speaking to them now with this answer to this question. Who are they? They're listening to this and they're saying, I'm doing this. I'm not aware of doing it. This is all news to me, Mike. You're saying I've been doing this my whole life and this defines me? This doesn't sound anything like me. So who are you? Who are you, Mike? And who is the person who's listening? What defines them?
Well, so a few things. So the fact that there are an incredible number of processes under the hood, it has been known for a really long time. So not only all the physiological stuff that's going on, I mean, you also don't have the experience of running your liver and your kidneys, which are very necessary for your brain function. You are also not aware of all the subconscious motivations and patterns and traits and everything else. So let's assume right now that whatever our conscious experience is, there is tons of stuff under the hood, not just the thing that I just said, but everything else that neuroscience has been studying for 100 years or more. There's lots going on under the hood, and that doesn't define you. It enables you to do certain things. It constrains you from doing certain other things that you might want to do. The hardware does not define you. I think the most important thing, and look, I think this is a really important question because I get lots of emails from people who say, I've read your papers. I understand that now I understand I'm a collective intelligence of groups of cells. What do I do now? I don't know what to do anymore, right? And my answer is do whatever amazing thing you were going to do before you read that paper. Like all of this information about what's under the hood is interesting, and it's it has all kinds of implications. But the one thing it does not do is diminish your responsibility for living the best, most meaningful life you can. It doesn't affect any of that. And one way I think about this, and there's lots of sci-fi about this, but one thing that you might remember is, you seen the film Ex Machina?
Yes.
And so there's one scene there where the protagonist, he's standing in front of a mirror and he's completely freaked out because the AI is so lifelike. He's now wondering maybe he's a robotic organism too. And so he's he's cutting his hand and he's looking in his eye and he's trying to figure out what he is. And so let's just dissect that for a minute. The reason he's doing this, and what happens to most people, which I think is quite interesting, is that if they were to open their arm and they find a bunch of cogs and gears inside, I think most people would be super depressed because I think where most people go with this is, I've just learned something about myself, meaning I know what cogs and gears can do. They're a machine. And I just learned that I'm full of cogs and gears. Therefore, I'm not what I thought I was. And I think this is a really unfortunate way to think, because what you're saying is your experience of your whole life and all of the joys and the suffering and the personal responsibility and everything else that you've experienced, you're now willing to give all that up because you think you know something about what cogs and gears can do. I would go in the exact opposite direction and I would say, amazing, I've just discovered that cogs and gears can do this incredible thing. Like, wow. And why not? Because why do you think that proteins and the ions and proteins and the various things in your body, those are great for true cognition. Like I always knew I was full of protein and lipids and ions and all of those things, and that was cool. I was okay with being that kind of machine. But cogs and gears, no way, right? And so I think one thing that we get from our education focused on certain kinds of materialism is that we get an idea, we get this unwarranted confidence in what we think different materials are capable of, and we believe it to such an extent. I mean, I find this amazing as a kind of an educational or sociological thing that. We imbibe that story so strongly that we're willing to then give up everything that we know about ourselves in order to stick to a story about materials. I think that's the one thing that I think Descartes had really right is that the one thing you actually know is that you are an agentic being with responsibility and a mind and all these and all this potential, and whatever you learn is on the background of that. So if you find out that you are made of cogs and gears, all you should conclude is, well, great, now I know this stuff can do it as well as proteins can. And so from that, so what I really hope that people get from this is simply the idea that pretty much no discovery about the hardware, no discovery about the biology or the physics of it should pull you away from the fundamental reality of your being, that whatever it is that you are, groups of cells, an emergent mind to pull down from Platonic space, or whichever of these things are correct. The bottom line is you are still the amazing integrated being with potential and a responsibility to do things.
So many people who dislike materialism and like the more, whatever they consider to be not materialism, so it could be idealism, it could be spiritualism, or whatever they want to call it, or non-dualism, or trialism instead of a dualism. In part, what they're saying is, look, I'm not material, because they denigrate the material and they view it as robotic, lifeless. But you're saying there's another route. If you are material, whatever it is you turn out to be, you can elevate that. So you can say, look, there's a dynamism to it. There's an exuberance. There's a larger than lifeness to what I previously thought was ossifying.
Yeah, for sure. And Ian McGilchrist makes a point of this too. He says that we've underestimated matter. When we talk about materialism, we have been sold and are selling still this notion of matter as lacking intelligence. And I think that we need to give up this unwarranted confidence in what we think matter can do. We are finding novel protocognitive capacities in extremely minimal systems, and they're surprising to us. They're shocking to when we find these things. And I think we are really bad at recognizing what kinds of systems can give rise to minds, and therefore being depressed because we think that we are a particular kind of system, and there's no way that system can be this majestic agentic being. It's way too early for that. We absolutely. I mean, this is one of the, I think, the major things is that we have this idea that we know what different kinds of matter can do, and obviously I'm not just talking about homogeneous blocks of material. I'm talking about specific configurations, but really minimal kinds of things can have some very surprising cognitive qualities. And so it's way too early to think that we know what's going on here.
I think we have a record here of 45 minutes of recording or so, and we haven't mentioned the word bioelectricity once. So kudos to you, kudos to me. How the heck did that happen? For a Michael Levin interview. So what does bioelectricity have to do with any of this?
Yeah. So bioelectricity is not magic. It is not unique in the sense that there are probably, this is certainly out in the universe, there are probably other ways of doing what it does. But what it does here on Earth in living forms is something very interesting. It functions as a kind of cognitive glue. So when you have collective intelligence, you need to have policies, and you need to have mechanisms for enabling competent individuals to merge together into a larger emergent individual that's going to do several things. First of all, the larger network, the larger level is going to distort the option space for the lower levels. So the parts are doing things that the parts do, but they're now doing it in a way that that is coherent with a much higher level story of what's going on, higher level goals, because their action landscape is bent. It's distorted by the larger level. Their perception, their energy landscape is distorted. So that collective is going to that new self is going to have memories, goals, preferences, a cognitive like that requires some very specific features, and there are a bunch of them. And one of the modalities that lets that happen, that lets the cognitive Lycone scale, that lets the intelligence scale, is bioelectricity. So by taking cells and enabling them to be part of an electrical network, there are some really interesting larger level dynamics, which, this is what we exploit in artificial neural networks, of course, right? This is biology noticed this since the time of bacterial biofilms, that electricity is just a really good way for higher level selves to emerge and higher level computation to emerge. There are probably other ways of doing it, but here on Earth, bioelectricity tends to be the way to go.
Something I always wonder in conversations about our higher selves, lower selves, higher goals. How do we even say higher or lower when what we're talking about is such a vast landscape of goals or cognitive light cones in a higher dimensional space, where the real number line is the only continuum that has an ordering to it? As soon as you have the complex numbers, or R2 or R3, etc., you can't pick 2 points and say one is higher than another.
Yep.
Unless you implement other structure. So what is it that allows us to say higher or lower?
Bad vocabulary. You're 100% right. The only thing that matters here, because it doesn't necessarily mean the next level up is not necessarily smarter than the lower level, usually, but that doesn't guarantee that at all. Not necessarily bigger or smaller in physical scale. So the only thing I mean by, and we really don't have a great vocabulary yet for all this stuff, but the only thing I mean by higher is something akin to set membership. Just the fact that a tissue is made up of cells, and cells are made up of molecular networks. That's it. That's all I'm talking about. I'm not saying that it's bigger or more intelligent or more valuable. All I mean is that in this hierarchy, certain things are made up of other things. That's it. That's all I mean.
Earlier, when defining intelligence, I believe you said William James's was something about ability, but also means. So ability to generate multiple paths to a single goal. I don't know if it was also the ability to have multiple goals, but we can explore that. But let's pick out a goal. Then you can generate multiple paths to that goal, many ways of executing. But then you also, I believe you said the means too as well. Is that correct?
Yeah, the means in James's, at least the way I read him, in James, when he says means, he means the path. That is the path. A means to an end, right? It's a path that takes you to that end. So, this is the kind of stuff we see in biology. Just to give you an example, one thing that people often think when they hear me talk about the intelligence of development and so on, people often think I mean the complexity, right? Just the fact that you start from an egg and you end up with, I don't know, a salamander or something, that there's an increase in complexity, and then rightly people think, well, that's just, there's lots of examples where simple rules give rise to complex outcomes. That's just emergent complexity. That's not intelligence. And they're right. That is not what I mean. That's not intelligence. What I mean by intelligence is the problem solving of the following kind. So let's say you have an egg belonging to a salamander. One of the tricks you can do is prevent it from dividing while the genetic material is copying, and so you end up with polyploid newts. So instead of 2n, you can have 4n, 5n, 6n, 8n, that kind of thing. Well, what happens when you do that? What happens is the cells get bigger in order to accommodate the extra genetic material, but the actual salamander stays the same size. So if you take a cross section, so let's say we take a cross section of a little tubule, a kidney tubule that runs to the kidneys. Normally, there's like 8 to 10 little cells that go in a circle to make that tubule, and then there's a lumen in the middle. So if you make the cells gigantic, the first thing you notice is that, well, first of all, having multiple copies of the genetic material, you still get a normal newt. So that's pretty amazing already. Second amazing thing is when the cells scale to the amount of genetic material, so the cells get larger. That's amazing. Then you find out, well, actually, since the cells are really big, only a few of them are now working together to make the exact same size tubule. So they scale the number of cells to make up for the aberrant size of the cell. Right? That makes sense. And then the most amazing thing of all happens when you make truly gigantic cells. There's not even room for more than one. One cell will bend around itself, leaving a lumen in the middle. The reason that's amazing is that requires a different molecular mechanism. That's cytoskeletal bending, whereas before you had cell-to-cell communication. And so that kind of thing, right? So just think about this. You're a newt coming into the world. You have no idea. You can't count on how much genetic material you're going to have, how many cells you're going to have, what size cells you're going to have. What you do have is a bunch of cool tools at your disposal. You have cytoskeletal dynamics, you have gene regulatory networks, you have bioelectricity, you have all this stuff. And what you're able to do under totally novel circumstances is pick from your bag of tools to solve the problem. I go from an egg to, in morphospace, I take this journey from an egg to a proper newt. Not only can I not count on the environment being the same, I can't even count on my own parts being the same, right? That kind of another way to call this attitude is beginner's mind. It's like you don't overtrain on your priors, on your evolutionary priors. You have a bag of tools, and you're not just a fixed solution. This is why I think evolution doesn't just produce solutions to specific environmental problems. It produces problem-solving agents that are able to use the tools they have. I mean, what's a better example of intelligence than something that can use the tools it has in novel ways to solve a problem it's never seen before, right? Like that is a version of intelligence, and that's what is all over the place in biology. The ability to navigate these pathways, not only to avoid various barriers and so on, but to use the tools available to them in creative ways to solve the problem. And we see some of this in extremely minimal systems. It does not require a brain, doesn't even require cells. Very minimal systems have surprising problem-solving capacities. And this is why we should be extremely humble when we try to make claims about what something is or isn't, or what competencies it has. We are not yet good at recognizing those things. We do not have a mature science yet of knowing what the properties of any of this stuff is.
So the tricky part with this definition of intelligence, help me out with this, is that what we want to say is that it's conceivable that the kid from Saskatoon, the poor kid, is more intelligent than the rich kid from the Bay Area. So that's conceivable. But the rich kid has far more means, far more ability to achieve their goals. So if there was implementability within the path, so if we say, look, the ability to generate paths that are realizable is in part what defines the IQ or the intelligence. Well, the poor kid from Saskatoon has less raw material to play with to generate a path. So how do we avoid saying, unless you want to say, which I don't imagine you want to say, how do we avoid saying that the poor kid from Saskatoon is just by definition less intelligent by happenstance than the person from the Bay Area?
Yeah. The thing to keep in mind here is that estimates of intelligence, and I think all cognitive terms, so everything about so all the words that people use, sentience, cognition, goal-directedness, all of them, I think we have to remember that those are not objective properties of a given system. IQ is not the property of whatever system you're trying to gauge the IQ of. It is your guess, your best guess about what kind of problem solving you can expect out of that system. So it's as much about you as it is about the system. And we've shown this in our experiments a lot of times, that when people talk about certain kinds of developmental constraints, or they talk about the competency of tissues to do one thing or the other, it's much more about our own knowledge of the right stimuli and the right ways to communicate with that system, and not so much about the system itself. When you make an estimate of the intelligence of something, you are taking an IQ test yourself. All you're saying is, this is what I know in terms of, and this is what I can see in terms of what kind of problem solving I can see. And this applies to animals, this applies to AIs, this applies to humans in various economic environments. The simple version of this is, you show somebody a human brain and they say, that's a pretty awesome paperweight, and I can see that it can do, least action and hold down my papers against gravity, and that's all I think it can do. And somebody else says, you've missed the whole thing, right? Like, this thing does all this other stuff. So I think that that type of mistake, where A, we think that it's an objective property of the system, and B, that we think that we're good at determining what that is what bites us a lot when we're dealing with especially unconventional systems. So to use your example, if someone looks at a kid with that environment and says, well, I don't think this kid has much intelligence, the problem isn't on the side of the kid. The problem is that somebody else might come along and says, oh, you don't get it. In a different environment, this kid would exhibit all these amazing behaviors. The good news about all of this, and I'm certainly not, it's not in my wheelhouse to comment on any of the kind of economic stuff or the sociology of it, but for the biology and for the computer science and so on, the good news is that all of these things are empirically testable. So when we come across a certain system, each of us is going to guess what is the problem space it's operating in, what are its goals, and what capabilities do we think it has to reach those goals, and then we do the experiment, and then we see who's right. That's the thing, that this is not a philosophical debate. This is absolutely experimental. So if you say, I don't think these cells have any intelligence, I think they're just a feed-forward emergent dynamics, and I think, oh no, I think they're actually minimizing or maximizing some particular thing, and they're clever about doing it. We do the experiment. We put a barrier in their place, between them and their goals, and we actually see, do they or do they not have what I claimed to be their competency, and then we find out how much each of our views lets us discover the next best thing. So these are all empirically testable kinds of ideas.
So before we get to consciousness and the Levin labs, I want to talk about cognition. So in 2021 or so, you had a paper called Reframing Cognition. Okay, something akin to that?
Yeah, that sounds like, I think that might have been a review with Pamela Lyon.
Yes,
maybe
yes.
That? Yeah.
And then on page 10, section 5, something like that, you started talking about basal cognition and uncaveated cognition. So what do those terms mean?
To be completely honest, I don't remember this part. I certainly don't remember the pages or the
basal cognition.
Yeah, so the idea for basal cognition is basically that whatever cognitive capacities we have, they have to have an origin, and we have to ask, where did they come from? Because this idea that we are completely unique and they suddenly sort of snap into place. It doesn't work evolutionarily and it doesn't work developmentally. Both of those are very slow processes. So the stories we have to tell are stories of scaling. They're to really understand these processes, we have to understand how simple information processing capacities scale up to become larger cognitive light cones, more intelligent systems, project into new problem spaces and so on. So basal cognition is the question of, okay, so where did our cognitive capacities come from? So that means looking at the functional intelligence of cells, tissues, slime molds, microbes, bacteria, and minimal matter, active materials, that kind of stuff. That's basal cognition. Well, what do the really primitive versions of cognition look like? And it's a really important skill to practice that kind of imagination, because often what trips people up is they imagine, for example, panpsychist views, right? So somebody says, "Oh, you're trying to tell me that this rock, is sitting there having hopes and dreams." Well, no, that's not the claim. The claim isn't that these full-blown, large-scale cognitive properties that you have are exactly there everywhere else. The claim is that it's a spectrum or a continuum, and that there are primitive, tiny versions of them that are also should be recognized, because we need to understand how they scale. So that's basal cognition.
So if it's a spectrum, I hear this plenty. Look, I'm not saying everything is conscious. It's a spectrum. It's not on off. But then to me, can't you just define on off to be if you have a non-zero on the spectrum, then you're on? Like, for instance, you say a particle has electric charge. Is it electrically charged? Well, it's on the spectrum. Yeah, if it has a non-zero amount, you call it electrically charged. If it's zero, then you say it's neutral. So can't you just say then that, yes, the rock does have hopes and dreams, even if it's at 0.00002% of whatever you have?
I personally am on board with that. I think potential energy and least action principles are the tiniest hopes and dreams that there are. So I agree with that completely. I think that is the most basal version. And in our universe, so this goes way beyond my pay grade, but for example, I've talked to Chris Fields, who is really an expert in this stuff, and I asked him, is it possible to have a universe without least action laws, right? And he said the only way you can have that is if nothing ever happens. So if that's the case, that tells me that in our world there is no zero on the cognitive scale, and everything is on it. But again, we have to ask then. So I agree with you. I think if you're on the spectrum, then you're on, and that's it. And I think in this universe everything is on. But we have to ask ourselves, what do we want this terminology to do for us? So that's why some people critique these kind of perspectives by saying, well, then if everything is on it, then the word means nothing. Then why do we even have the word? Because if everything is cognitive, and I didn't say consciousness yet, but let's say everything is cognitive, then why do we need the word everything? And I really think that we need to focus on what we expect the terminology to do for us. So let's imagine, let's just dissect this for a minute. The old paradox of the heap, right? So you got a pile of sand, and you know that if you take off one little piece of sand, you still got a pile, but eventually you have nothing. So how do you define the pile? So I think for all of these, so my answer to this, and I think the solution to all of these kinds of terminological issues, is that it's not about the object itself, it's about what tools you're going to use to interact with it. So if you call me and you say, I have a pile of sand, all I want to know about the definition of pile is, am I bringing tweezers, a spoon, a shovel, a bulldozer, dynamite? How are we, like, what are the tools that I have to do what we need to get done? So that is the only value in this terminology. So, by saying that everything is cognitive, does that by itself help us with anything? No. I think what does help is if you tell me what kind of cognition and how much, and that's an empirical question, and then we can argue about it. And the answer to that question is, what are the tools that help us the most? So you show me a bunch of cells and you say, I think this the right way to do this is physics, chemistry, and feed-forward emergence and complexity. That's how I think we're going to interact with it. And I look at it and say, I think the way to interact with this is through some interesting concepts from cognitive neuroscience, including active inference, learning, training, and so on. Then we get to find out who's right. If I can show that using my concepts I got to new discoveries that you didn't get to, well, there you go. On the other hand, if I waste all my time writing poems to a rock and nothing ever comes of it, well, then you're right. And so I think that the point of all this terminology, yes, we can say it's all on a spectrum, but now comes the fun and interesting work of saying, okay, so what does the spectrum look like, and where on the spectrum do the various things that we see land?
Okay, let's get to consciousness. I want to say that I don't agree with Chris Fields about the principle of least action, because firstly, people say the universe is lazy, but you can also put a minus sign and say the principle of maximum effort. Okay, but then also there are many quantum field theories that aren't based in Lagrangians to minimize. So there's algebraic, constructive, axiomatic, and categorical. And then there's a new video that actually got released a couple of days ago by Gabriel Carcassi. And I'll put the link on screen, which says there's a distinction between Newtonian, Lagrangian, and Hamiltonian mechanics. So Hamiltonian is more about flows. You just watch the flow of the system. Lagrangian is the one where you minimize. And then Newtonian, there's actually some Newtonian systems, F equals ma, that you can't map to a Hamiltonian system. So I have a bone to pick with Chris Fields.
You should have him on. This is getting way beyond anything I could argue with you about, but you should have him on and you guys could talk. I would watch that for sure.
We had a three-way discussion, again, a plug here, with Michael Levin, Carl Friston, and Chris Fields. That was fun.
Yeah.
Okay, so many people want to know, what is your hunch at which... See, there are various interpretations of quantum mechanics. We're not going to go there, but there are various theories of consciousness in the same way. There's a litany. Which one do you feel like is on the right track?
Well, let's see. I can say a few things. What I definitely don't have yet, I'm working on it, but I don't have anything that I would talk about now, a new theory of consciousness. So I do not have anything brilliant to add to this that somebody else hasn't already said. So I'm just going to kind of tell you what I have to say now.
Sure.
I think that one thing that's really hard about consciousness, and what makes it the hard problem, is that unlike everything else that we work with, we don't have any idea what a correct theory would output. So what format would the predictions
Sorry, we don't have an idea of what the correct theory would look like?
No. We don't have a correct. We don't have any idea of what the output of a correct theory would look like. What would it give you, right? So for everything else, the correct, a good theory gives you numbers, predictions about specific things that are going to happen. What does a good theory of consciousness give you? So, what we would like is something that, we say, okay, here's a cat, or here's a cat with 3 extra brain hemispheres grafted on that also has wings. What is it like to be these creatures, right? What is the output of a correct theory of consciousness? Because if it outputs patterns of behavior or physiological states, then what you've explained is physiology and behavior. There are going to be people that say, well, you haven't explained the consciousness at all. In fact, almost all theories of consciousness look kind of eliminativist, even the ones that aren't trying to be, even the ones that say, no, we're not trying to explain away consciousness, it's real, and I'm going to explain it. Then you look at the explanation and you always feel like, yeah, but you haven't explained the actual, the actual consciousness. You've explained some kind of behavioral propensities, physiological states, or whatever. So that's the problem that we have. Consciousness is one of those things that cannot exclusively be studied in the third person. Everything else you can study as an external observer, and you don't change much as the observer by studying them. Consciousness, you can really only, in a full way, you can only study consciousness by being part of the experiment, by experiencing it from the first person perspective. So the weak version of this is, you might say, well, a good theory of consciousness is art. What it outputs is art, poetry, whatever. That when we experience it, it makes us experience that conscious state. And we say, oh, so that's what it's like. I see, right? So that's one. But that's kind of a weak form. You can do a stronger form, and you say, well, the real way to do it is to have a rich brain interface. So if I want to know what some other system is what its consciousness is like, we need to fuse together. Now, caveat to that is you don't find out if you do that. So let's say a rich kind of brain interface, we really connect our brains together or something. You don't get to find out what it's like to be that system. Both of you find out what it's like to be a new system composed of the two of you. So it's still not. So from that perspective, it's really hard. I mean, people who do meditation or take psychedelics, I suppose they're doing experiments in actual consciousness. But third person experiments in consciousness are really hard. You can do things like turn it off, so there's general anesthesia, and you can say, oh look, the consciousness is gone. And even then, some people will say, yeah, but I experienced floating above my body while you did the surgery, and I saw you drop the scalpel and do this or that. So it's still, even with that amazing reagent of being able to supposedly shut off consciousness, you still get some issues. So the study of consciousness is hard for those kinds of reasons. And I think that about the only useful thing I could say here is that for the same reasons that we associate consciousness with brains, for exactly those same reasons, we should take very seriously the possibility of other forms of consciousness in the rest of our bodies, and also lots of other things. But so I'm working. So Nick Rouleau and I are working on a paper on this, where you can sort of look at all the different popular theories of consciousness on the table, and you can just ask which of them are specific for brains and why. Like what aspects of each of those theories really tells you that it's got to be brains. My guess is, we haven't finished the paper yet, but my guess right now is that there's not a single one that can distinguish brains from other types of structures in your body. And so I think we should take very seriously the possibility that other subsystems of the body have some sort of consciousness. It's not verbal consciousness.
Sorry, I'm not understanding. Are you saying, what if we list all the theories of consciousness and then we place, does it distinguish the brain as being responsible for consciousness?
Yeah, we ask what is it about that theory that says it's in the brain rather than somewhere else? Let's say your liver.
So IIT would say no, because IIT is a panpsychist theory that would say, look, if your liver is doing some processing, then it has some non-zero amount of consciousness.
Right, and I agree with that. Now, as far as I understand, IIT also has an exclusion postulate that says there should only be one central consciousness in the system. I think that's true. At least it used to be true. I don't know. Julia may disagree with that, but I think we are actually a collection of interacting perspectives and interacting consciousnesses for that reason. And then sometimes people say, well, I don't feel my liver being conscious. Right, you don't, but you don't feel me being conscious either. Of course you don't. And the fact that your left hemisphere has the language ability for us to sit here and talk about it, and the liver doesn't actually mean that it's not conscious. It just means that we don't have direct access to it, and we don't have direct access to each other. So that doesn't bother me. That's my suspicion about consciousness, is that for the same reason that people think it's in the brain, we should take very seriously that it's in other places in the body. And then more generally, other types of constructs that are not human bodies at all, or not even animal bodies.
You've spoken to Bernardo Kastrup several times now. What is it you agree with him the most that you think most people would disagree about? Because you agree with him that it's nice to go for a walk. Okay, sure, but most people agree it's nice to go for a walk. So what is it that you agree with him about that you think is a contentious issue to most people? So this is a controversial statement. And then what is it you disagree with him about regarding consciousness?
Yeah, boy, I don't, it's hard for me to know what most people agree or disagree with him about. I really don't know. We agree on a lot of things. We agree on, I think, the primacy of consciousness. I think that, his idealist position has a lot to recommend it. The one thing I think we disagree on is the issue of compositionality. So if I recall correctly from a talk that we had together a little while ago, he felt that it is important, in order to be a true self, to have a conscious experience and an inner perspective. You have to be, he focuses on the view of embryonic development as a single system that, whatever subdivides and develops, but it starts out as a single system. And I was arguing that really is just a contingent feature of biology. I mean, we certainly can take two early embryos and mush them together. You get a perfectly normal, embryo out of it. And in general, there are lots of biological systems like our xenobots, like anthrobots, that you can create by composition, by pulling other things together. So I don't give as much, I don't put as much emphasis on a system being demarcated from the outside world because it was somehow, because it started out that way and it sort of remained, disconnected. I think I think that's kind of a superficial aspect of the biology, and you can do things a different way. I don't think that's what's responsible for, but I think he thinks it's important that individual selves are not compositions. They're not made as compositions. They're somehow individualized from the word go. Which again, even the egg, right? So we humans like eggs because we can see it as a distinct little thing with a membrane. You say, there's an individual. But even an egg is composed by the maternal organism from molecular components. Like I see no point at which any of this is truly distinct from anything else. So I put less emphasis on it, but I think he thinks it's important.
It seems like the point that you're saying is, look, we can think about this as several rooms. This building comprises several rooms, but and Bernardo may say that's what makes a person is the distinct rooms. But you're saying, yeah, but even in a room, there are different people, there are different chairs, there are different tables. Is that what you're saying?
What I'm saying is, and I may not be doing justice to his view, and I think you should ask him more about this, but I think he thinks that it's important in order to be a unified. So I think we were discussing what makes for a unified inner perspective, right? So we don't feel like billions of individual brain cells. I mean, I have no idea. Well, we kind of do because that's what it feels like to be billions of individual cells of neurons. That's really what it feels like. But we do feel, at least most of us, most of the time, feel like some kind of unified, centralized inner perspective. And so we were talking about how that comes about. And I think he felt that having that in the physical universe is importantly related to arising from a single origin. So he sees the egg as a single point of origin, and arising from that's how you are a separate individual from others. And I see it as much more fluid, and I see the boundary between self and world as something that can change all the time. I think it changes in embryogenesis, and that's the story of the scaling of the cognitive icon that we talked about. I think it can shrink during cancer. I think it can change during metamorphosis, during maturation. I think it's much more fluid than that.
Now, as we're on speculative ground, if what makes an agent is the distinction between the self and the world, and some people think of God as the entirety of everything, thus the entire world, and there's no distinction, then can one say that God is an agent?
I don't know. I mean, certainly, I think most religions that have a God, anyway, as far as my understanding is, they would think that yes, that God has extreme agency, in fact, higher than ours. I don't know what that really buys us, in any helpful way.
Remove the word God. Does the world have agency?
Okay, so that's an interesting question. So let's start with, first of all, how do we know when anything has agency? And that's an experimental research program. So you hypothesize what problem space it's working in, what you think its goals are, and then you do experiments to figure out what competency it has. And then you find out, did I guess well, poorly? Do I need to make a better guess, and so on? So for example, people have said to me, well, your kind of panpsychist almost view says that the weather should be cognitive. And I say, I don't say that it is or isn't because we haven't done the experiments. Do I know that weather systems, let's say hurricanes or so on, do I know that they don't show habituation, sensitization, that they couldn't be trained if you had the right scale of machinery? I have no idea. But what I do know is that it's not this is not a philosophical thing that we can decide arguing in an armchair. Yes, it is. No, it isn't. No, you have to do experiments, and then you find out. So now the question is, okay, so what about the galaxy? What about the universe? Right? Are these the Gaia ecosystems? Again, I think these are all empirical questions. Now, some of them are intractable. We don't have the capability to do experiments on a planetary scale. But for example, one thing that I did try to do once was design a gravitational synapse. So design a solar system size arrangement where masses would fly in, and based on the history of masses flying in, it would respond to new masses in a different way. So you can do historicity, and you can have habituation and sensitization and things like that. So could you have something like that would be very sort of ponderously, slowly, on an enormous scale, computing something and having sort of simple thoughts? I bet you could. I bet you could. Is the real universe doing that? I have no idea. We have to do experiments. So here you bump up against another question, which is, how do you know if and when you are part of a larger cognitive system, right? So I don't, how do we know if we are in fact part of a bigger mind? So I don't know. And my suspicion is that there's some sort of Gödel-like theorem that will tell you that you can never know for sure, and you can never be certain. But I bet that you could gather evidence for it, for or against. And I often think about a kind of mental images. Imagine two neurons in the brain, and one is a kind of a strict materialist, and one's a little more mystical. And the one neuron says, look, we just run on chemistry, and the outside world is a cold mechanical universe, and it doesn't care what we do. There's no mind outside of us. And the other one says, can't prove it, but I kind of feel like there's an order to things, and I kind of feel like our environment is not stupid. I kind of feel like our environment wants things from us, and I kind of feel these waves of these waves back propagating through us that are like almost rewards and punishments. I feel like the universe is trying to tell us something. And the first one says, you're just seeing faces in clouds. It doesn't exist. And of course, in my example, the second one is correct, because they are in fact part of a larger system. They're part of a brain that is learning things, and it's very hard for any one node in that system to recognize that, or even a sub network. But I wonder if we could, having a degree of intelligence ourselves, if we could gain evidence that we were part of a larger system that was actually processing information. And I don't know exactly what that would look like, but my hunch is that it would look like what we call synchronicity. I think that what it would look like are coincidence are events that don't have a causal connection at our lower level, like mechanistically, like by physics. There's no reason why that should be, but at a larger scale in terms of meaning and the greater meanings of things, that they do have some kind of interpretation. And I think that's what it would look like to be part of a larger system. I think it would look and feel like synchronicity. So does it exist? I don't know, but that's what I think it would feel like.
Take me through the history of the Levin lab. When did it start?
Well,
What were your what were your first breakthroughs?
Yeah. Okay. Let's see. Well, it started. I mean, it started in my head when I was pretty young. Like it was a dream that I had to do this kind of stuff. I mean, I consider myself to be the luckiest person in the world. I get to do the funnest stuff with the best people. So I think it's super fortunate. But I kind of had this idea when I was very young. I had no idea what it was like. I was pretty sure that it was actually impossible. I never really thought it would be practically feasible, but I figured I would push it as far as I could before I would have to go back to coding and because
For people who are unaware, your background's also in computer science.
Right. Yeah. I learned to program pretty young, and at that time, that was a pretty good way to make money. And I just figured I would do the biology as long as I could, and then eventually I would get kicked out, and then I would just go back to coding. So yeah, my lab actually began in September of 2000. That's when I got a faculty position at the Forsyth Institute at the Harvard Medical School, and we opened our doors in 2000. It was just me at first, and then me and one other technician. There's like 42 of us now, but at the time it was just me and a tech named Adam. That was the first time, starting then was the first time that I could really start to take practically to be practical about some of the ideas I had about bioelectricity and cognition and all these things. Prior to that, I was building up a tool chest, so I was building up skills, techniques, information, and so on. But being a grad student and then a postdoc, I wasn't able to talk about any of these things. But then when I was on my own, and then that was the time to get going. So just a couple of interesting milestones. Already by the time I got we opened the lab, I was involved in a collaboration with Ken Robinson and his postdoc Thorley Thorlin, and together with my postdoc mentor Mark Mercola, we really showed the first molecular tools for bioelectricity. So we had a paper on left-right asymmetry, and we showed the first bioelectric tracking of non-neural bioelectric states in the chicken embryo. We showed that it was important for setting up which side is left, which side is right, and then manipulating that information using injected ion channel constructs. So that was the first time any of that, reading and writing the mind of the body, which is how I certainly wouldn't have said it back then, but this is how I see it now. That was the first time that was done in a molecular way. So that cell paper came out in I think 2002. I think it finally came out. But that was a really early project. The other really early project was I had as a postdoc, I started gathering tools for this whole effort, and a lot of those tools were DNA plasmids encoding different ion channels. And so what I would do is I would write send emails or letters to people working in electrophysiology, gut physiology, inner ear, and they would have some potassium channel that I cloned, and I would say, could I get one of these plasmids? And I was telling them what I was going to do. I would say, and what I'm going to do is use it to express it in embryos in various locations, and use it to in a very targeted way change the bioelectrical properties of these things. And most people were very nice, and they sent me these constructs. One person sent a letter to my postdoc mentor to say that I had clearly had a mental breakdown, and that he should be careful because this is so insane that I'm obviously off my rocker, right? And so I remember.
No, wait, is this your recapitulation of what they said, or they actually said mental breakdown?
No, well, okay. So I didn't see the letter, but my boss came to me. So my boss came to me, and he was laughing, and he said, look at this. This guy says you're nuts. You asked him for a plasmid. He told me to watch out. He says you're having a psychiatric break. So that's what I'm relaying is what he said to me.
Okay.
So but nevertheless, most people sent constructs, and when we got to the lab, when my lab opened, I started doing that. I started misexpressing these things in embryos to just to see the space of possible changes. Right? What does bioelectricity really do? I mean, nobody knew at the time. It was thought it was really crazy. It was thought that membrane voltage was a housekeeping parameter. There was an epiphenomenon of other things that cells were doing, and that if you mess with it, all you're going to get is uninterpretable death. That's everybody thought this was a stupid idea. And so we started doing this. And I had this graduate student. She was in the dental program. Her name was Ivy Chen, and she was in the dental program. And she had amazing hands. And so I taught her to micro-inject RNA into cells in the embryos because she had like really good hands.
How do you know she had good hands before she tried that?
Well,
you look like you have good hands.
Yeah, well, she was a dental student, and so I talked to her. She wanted to do research, and I said, "Tell me what you do." And they said, "Oh, I do these. I sew up people's gums and whatever." And I said, "Okay, you probably could do this."
Okay, so she wasn't playing Call of Duty.
No. She may have been also, but I don't know.
All right,
I don't know that. What I know is that she was doing surgeries in people's mouths, and I thought that she may be able to in tight confined places with the glasses and everything. So I thought she would be able to do this through a microscope, and she was. And so we did this together, and we injected these constructs. And I still remember to this day. She calls me in one day, and she says, "So I looked at the embryos, and they're covered with some kind of black dots." And I said, "Black dots. Let me see. Let's go look." So I come out and we look through the microscope. The black dots were eyes. What she had done was the potassium channel that she injected was the one that we later took years to publish the paper. But what she had discovered is a particular bioelectrical state that tells cells to build an eye. It's remarkable because right there and then you knew that A, bioelectricity was instructive. It was not an epiphenomenon because it controls which organs you get. B, that the whole system is modular and hierarchical because we did not tell the cells how to build an eye. So we didn't say where the stem cells go or what cells go next to what other cells or what genes should be expressed. We did none of that. We triggered a high-level subroutine that says build an eye here. So right away, that one experiment like told us all these amazing things. Then eventually, and this was the work of a grad student, Sherry Au, in my group, who took on the project, and she did a whole PhD on this, showing that the other amazing thing about it is that if you only target a few cells, what they do is they get their neighbors to help out because they can tell there's not enough of them to build an eye. Kind of like ants. Ants recruit their buddies to take on a bigger task. So that tells you that the material you're working with has these amazing properties that you don't have to micromanage. Right? It's a different kind of engineering. It's engineering, as I put it recently in a recent paper. It's engineering with agential materials because it's a material with competencies with an agenda. You don't have to control it the way you do wood and metal and things like that. So okay. So anyway, so that kind of thing. Then we had a bunch more work on left-right asymmetry and showing how the cells in the body decide which side they're on based on these electrical cues. Then we discovered that in order for cells to the way they interpret these electrical cues had to do with the movement of serotonin. So long before the nervous system or the brain shows up, the body is using serotonin to interpret electrical signals. So this was really underscoring the idea that a lot of the tools, concepts, reagents, pathways, mechanisms from neuroscience really have their origin much earlier. And so this was a completely new role for serotonin, right? So serotonin is a neurotransmitter, does many interesting things, but long before your brain appears, it also controls which direction of your body, your heart, and your various other organs go to. So trying to understand in the kind of, in the short term, how does electrical activity control cell behavior, but bigger picture, wow, these neural-like processes are going on in cells that are absolutely not neurons, much more, much long before that. So that was kind of cool. Then I hired a postdoc named Danny Adams, who later became a faculty member and a colleague. And one of the things that she did was to pioneer the early use of voltage-sensitive dyes to read these electrical potentials. And so she discovered in the work that she did in my group, she discovered this thing we call the electric face.
And approximately what year is this now?
Oh, this is the electric face. This is probably 2008, something like that, 2007, 2008. And what she had discovered was that if you look at the nascent ectoderm that later will regionalize to become face and mouth, eyes and mouth and all of that, early on, before all the genes turn on that determine where all those things will go, the bioelectric pattern within that ectoderm looks like the face. It shows you where all this stuff is going to go. And then we were able to show that. And by the way, there was that eye spot, which is why the eye thing worked. And we were able to show that all kinds of birth defects that mess up the formation of face do it by inhibiting the normal bioelectric pattern, and that you could fix it. You could exert repair effects that way. So that was interesting. Then we started looking at regeneration. And again, the early work was done also by Danny, and then later on by Kelly Chang, who's now a faculty member at University of Las Vegas. Where what we did was we showed that tadpole tail regeneration was also bioelectrically driven, and that was our first gain of function effect in regeneration, where we were able to show that we could actually induce new regeneration. So the tail is a very complex organ. It has spinal cord, muscle, bone, not bone, vasculature, innervation, peripheral innervation, skin. And so we took tadpoles that normally do not regenerate. There's a stage at which they can't regenerate their tails, and we developed a bioelectric cocktail that induces it to grow. My postdoc at the time, Kelly Chang, said I soaked them. I said, well, how long did you soak them for? And she said an hour. And I thought, but that's got to be too short. There's no way an hour soak is going to do anything. And sure enough, that hour soak led to 8 days of regeneration where we don't touch it at all. And the most recent version of that work is for the frog leg, where we show that 24-hour stimulation with our cocktail induces a year and a half of leg growth, during which time we don't touch it at all. So the amazing thing there is, again, this is not micromanagement. This is not 3D printing. This is not us telling every cell where to go during this incredible year and a half long process. This is at the very earliest moment you communicate to the cells, go down the leg building path, not the scarring path, and that's it. And then you take your hands off. It's calling a subroutine. It's modularity. It's relying on the competency of the material, where you're not going to micromanage it. So that was the first time that kind of became obvious that was possible, is when she showed that just an hour stimulation of the correct bioelectrical state got the whole tail to commit to regenerate itself. So that was the beginning of our regeneration program, after which we went into limbs, and now of course we're trying to push into mammalian limbs. Along the way, Celia Herrera Rincon and Naroshia Murugan were other postdocs that showed leg regeneration in frog and so on. Around that time, we had another thing. I really wanted to work on cancer, and I really wanted to work on this idea that there's a bioelectric component to it, and the way you can think about it is simply that not why is there cancer, but why is there anything but cancer? So why do cells ever cooperate instead of being amoebas? Why do they ever cooperate? And so we know that the bioelectric signaling is the kind of cognitive glue that binds them together towards these large-scale construction projects, maintaining organs and things like that. And so, so we wanted to study that bioelectrically. And so I had 2 students, Brooke Chernet and Maria Lebikin, who undertook that. And we were able to show that using this bioelectrical imaging, you can tell which cells were going to convert ahead of time. You could also convince perfectly normal cells to become metastatic melanoma just by giving them inappropriate bioelectric cues about their environment. So you can no genetic damage, no carcinogens, no oncogenes, but just the wrong bioelectric information, and they become like metastatic melanoma. And best of all, they were able to show that you can actually reverse reverse a carcinogenic stimuli, for example, human oncogenes, by appropriate bioelectric connections to their neighbors. So we had a whole set of papers showing how to control cells bioelectrically. And Juanita Matthews in my lab now is trying to take all those strategies into human cancer.
So this is 2009?
This was, the first experiments were done around 2010, 2011, something like that.
So when did this conjectural connection between bioelectricity and cancer occur to you? The field was clueless about that. It's not as if they had an opinion and said no.
Well, to be clear, the very first person who talked about this was Clarence Cohn in 1971. So Clarence Cohn in 71 wrote that he had a couple of papers in Science where he showed that resting potential of cells was an important driver of cell proliferation, and he conjectured that it might have something to do with cancer. So this idea had been floated. Nobody had ever done anything with it, and the tools to study this at a molecular level didn't exist until we made them. So that that idea, just that bioelectricity is important in cancer, had been around before. What I think we brought to it that was completely new is the notion of that this is also related to cognition. The idea that it's not just that it drives proliferation in cancer, that this is really involved in limiting the size of the cognitive light cone, at which point cells acquire ancient metastatic-like behavior, the way that amoebas do. That aspect of it, I think, is completely new with us. That idea that this really is about the boundaries of the self. That I've never seen anybody else talk about that. So those around that same time, there's something else interesting that happened in 2009, which is that we were studying planaria, we were studying flatworms, and we had shown that when you cut planaria into pieces, the way that these pieces decide how many heads they were going to have is actually related to the ability of cells to communicate with each other using gap junctions, these electrical synapses. And so we had made some two-headed worms and so on. And around 2009, I had this student, this visiting student from BU. Her name was Larissa, and I asked her to recut the two-headed worms. And just in plain water, no more manipulation of any kind. We
recut them, meaning cut off the heads of them?
Again, yeah. So you have a normal one-headed worm. You cut off the head and the tail. You have the middle fragment. You soak that middle fragment in a drug that blocks the cells from electrically communicating with each other, and they develop heads at both ends.
Would that drug that you soak it in be called a biological cocktail? Like, is that what you referred to earlier? It's a different biological cocktail.
It wasn't even a cocktail. It was one single chemical. It was really simple. It was just one single chemical, and all it does is block gap junctions. And so what that did was change the electric circuit properties that the cells have, and both wounds decided they had to be heads. So now you get these two-headed worms. So Larissa recuts these two-headed worms in plain water. No more manipulation, and she gets more two-headed worms. It's permanent. Once you've convinced them. Now the genetics are untouched, right? No genomic editing, no transgenes. It's genetically identical, but the two-headed worms are a permanent line now. So a couple of interesting things there. One is one is it shows the interesting memory properties of the medium, meaning once you've brought it to a new state, it holds. It remembers the it's a kind of memory. It remembers the two-headed state. Another interesting thing is that two-headed worms were first seen. Well, they were first described in the early 1900s. So people had seen to be made by other means. People had seen two-headed worms. Apparently, to my knowledge, I don't think anybody's ever written about this. Nobody thought to recut them until we did it in 2009. And I think the reason is because it was considered totally obvious what would happen. I mean, their genome is normal. You cut off that second ectopic head, and of course, it'll just go back to normal. That's what people assume. So this is another example of why thinking in these different conceptual ways is it matters. It leads to new experiments, because if you don't think about this as memory, if you're focused on the genes as driving phenotypes, then you would never. It doesn't make any sense to recut them. But if you start thinking, well, I wonder if there's a physiological memory here, then that leads you to this experiment, right? So thinking in this way leads to new experiments. And then the other thing it points out is something really interesting. So for pretty much any animal model, you can call a stock center and you can get lines of genetic mutants. So you can get flies with curly wings and mice with crooked tails and weird coat patterns and chickens with funky toes. So you can get any kind of mutant lines. In planaria, there are no mutant lines. Nobody's ever succeeded in making anything other than a normal planarian, except for our two-headed form, and that one's not genetic. And so there's a deep reason, which I didn't understand back then. In fact, I think I only really figured out what I think it means in the last few months. But it was striking that the only unusual planarian form, permanent planarian form out there, was the one that we had made, and that's the one that's not genetic. It's not done by the way that you would do this with any other animal. Yeah.
What's your recent discovery been?
Well, it's not so much a discovery. It's more of a new way of thinking about it. So one of the weird things about planaria is that because the way they reproduce, at least the ones that we study, the way they reproduce is they tear themselves in half, and then each half grows the rest of the body. And typically what happens for most of us that reproduce by sexual reproduction is that when you get mutations in your body during your lifetime, those mutations are not passed on to your offspring, right? They disappear with your body, and then the eggs go on and so on. Well, in planaria, it's not like that. In planaria, any mutation that doesn't kill the cell gets expanded into the next generation, because each half grows the remainder of the body. And so their genome's very messy. They have, in fact, cells can be mixoploid. They can have different numbers of chromosomes. That's very weird. And I always thought, isn't it strange? And nobody ever talked about this in any biology class that I've ever had. Isn't it strange that the animal that is the most regenerative apparently immortal, they don't age, cancer resistant, and by the way, resistant to transgenes, so nobody's been able to make transgenic worms, is also the one with the most chaotic genome. Now that's bizarre, you would think, but from everything that we were told about genomes and how they determine phenotypes, that the animal with all those amazing properties should have really pristine hardware. You would think that you should have a really clean, really stable genome if you're going to be regenerative and cancer resistant and not age and whatever. It's the exact opposite. I always thought that was incredibly weird. And so finally, I think, and we've done some computational work now to show why this is. I think we now understand what's happening. What I think is happening is this. Imagine, let's go back to this issue of developmental problem solving. So if you had a passive material such that you've got some genes, the genes determine what the material does, and so therefore you have an outcome, and that outcome gets selected, either does well or not, and then there's differential reproduction. So the standard story of evolution. Then everything works well, and everything works like it would in a genetic algorithm. Very simple. The problem with it, of course, is that it takes forever because let's say that you're a tadpole and you have a mutation. Mutations usually do multiple things. Let's say this mutation makes your mouth be off kilter, but it also does something else somewhere else in the tail, something positive somewhere in the tail. Under the standard evolutionary paradigm, you would never get to experience the positive effects of that mutation because with the mouth being off, you would die, and that would be that. So selection would weed you out very quickly, and you would have to wait for a new mutation that gives you the positive effects without the bad effects on the mouth, right? So that it's very hard to make new changes without ripping up old gains and so on. So that's some of the limitations of that kind of view. But a much more realistic scenario is the fact that you don't go straight from a genotype to the phenotype. You don't go from the genes to the actual body. There's this layer of development in the middle, and the thing about development is not just that it's complex, it's that it's intelligent, meaning it has problem-solving competencies. So what actually happens in tadpoles is if I move the mouth off to the side of the head, within a few weeks, it comes back to normal on its own, meaning it can reach again that region of anatomical space where it wants to be. So now imagine what this means. Imagine what this means for evolution when you're evolving a competent substrate, not a passive substrate. By the time a nice tadpole goes up for selection to see whether it gets to reproduce or not, selection can't really see whether it looks good because the genome was great or because the genome was actually so-so, but it fixed whatever issue it had. Right? So that competency starts to hide information from selection. So selection finds it kind of hard to choose the best genome because even the ones with problems look pretty good by the time it's time to be selected. So what happens, and we did computational simulations of all this, what happens is that when you do this, evolution ends up spending all of its effort ramping up the competency because it doesn't see the structural genes. All it sees is the competency mechanism. And if you improve the competency mechanism, well, that makes it even harder to see the genome. And so you have this ratchet, you have this positive feedback loop where the more competent the material is, the harder it is to evolve the actual genome. All the pressure is now on the competency. So you end up with kind of like a ladder, really an intelligence ratchet because, right? And people like Steve Frank and others have pointed this out for in other aspects of biology and also in technology, right? Once RAID array technology came up, it became not as important to have really pristine and stable disk media because the RAID takes care of it, right? So the pressure on having really stable disk is off. So what it means is that in the case of the planaria, that positive feedback loop, that ratchet went all the way to the end. That basically what happened here is that what you've got is an organism where it is assumed that your hardware is crap. It's assumed that you're full of mutations. All the cells have different numbers of chromosomes. We already know the genetics are all over the place, but all of the effort went into developing an algorithm that can do the necessary error correction and take that journey in morphospace no matter what the hardware looks like. That is why they don't age. That is why they're resistant to cancer, and that's why nobody can make a transgenic worm because they really pay less attention to their genome in that sense than many other organisms. So you can imagine a sort of continuum. So you've got something like C. elegans, the nematode, where they're pretty cookie cutter. So as far as we know, they don't regenerate much. It's what you see, what the genome says is pretty much what you get. Then you've got some mammals, right? So mammals, at least in the embryonic stages, they've got some competency. You can chop early mammalian embryos into pieces and you get twins and triplets and so on. Then you get salamanders. Salamanders are they're quite good regenerators. They're quite resistant to cancer. They're long lived. And then when you run that spiral all the way to the end, you get planaria, which are these amazing things that have committed to the fact that the hardware is going to be noisy and that all the effort is going to go into an amazing algorithm that lets them do their thing. And that's why if you're going to make lines of weird planaria, targeting the structural genome is not helpful. But if you screw with the actual mechanisms that enable the error correction, aka the bioelectricity, that's when you can make lines of double-headed and so on, because now you're targeting the actual the problem-solving machinery.
And if you were to look at the genome of the salamander versus the C. elegans, would the C. elegans be more chaotic or more ordered than the salamander?
It's a good question. So nobody's done that specifically. No, as far as I know, this is something that we're just ramping up to do now. Is to start.
Because correct me if I'm incorrect. It sounds like the hypothesis is that if you have a large amount of genetic chaos, or if you can quantify that, then you would have something that would be compensated for in terms of competency or some higher level structure.
Yeah, I think that's a prediction of this model that I just laid out. And so so we can test that. I mean, part of it also is there's an ecological component. I mean, you can ask the question, so why doesn't everything end up like planaria? And I think there's an aspect to this that ratchet obviously doesn't run to the end in every scenario, because in some species there's a better trade-off to be had somewhere else.
I wonder if there are three components then, because then if you don't see a direct correlation, it could be hidden by a third factor.
Yeah. Which I think would probably be environment. It would probably be the ecology of how do you reproduce, how noisy, how dangerous, how unpredictable is your environment. I'm going to guess there's something like that involved here. Yeah, but I think that's that's starting to kind of explain what's going on with planaria, and so we found we found the persistence of the two-headed phenotype, and then Nestor Oviedo and Junji Morikuma in my group wrote a nice paper on that, and so on. And the next kind of big advance there in 2017 was by Fallon Durant, a grad student in my lab who also did something interesting. So when you take a bunch of worms and you treat them with, let's say, this reagent that blocks the gap junctions, typically what you see is, okay, you treat 100 worms, 70% of them go on to make two heads, and 30% are unaffected. So we thought they were unaffected because they stay one-headed, and we always call them escapees because we thought that they somehow just escaped the action of the octanol. Maybe their skin was a little thicker or something, and we never had a good explanation for it. But anyway, there was a 70% penetrance to the phenotype, and most things have not 100% phenotype, so it wasn't.
Penetrance?
Penetrance just means that when you apply some treatment to a population, not all of them have an effect, and not all of them have the same effect. That's true for pretty much every drug, every mutation, and so on. So for years we called them escapees, and then around 2015, when Fallon joined the lab, she recut some of those one-headed escapees and found that they also do 70-30, that 70% of them became double-headed and 30% didn't. And so what we realized was that they're not actually escapees. They're not unaffected. They're affected, but the way they're affected is quite different. They are randomized. They can't tell if they should be one-headed or two-headed, and they flip a coin set with a 70-30 bias about what they should do in any given generation. In fact, we were able to show that when you cut multiple pieces from the same worm, and we call them cryptic worms, cryptic because physically they look completely normal, one head, one tail. They look normal, but they're not normal, and because if you recut them, they're not sure what to do. Their memories are bistable. So what happens is that you can cut them into pieces, and every piece makes its own decision if it's going to be one-headed or two-headed, even though they came from the same parent organism with the same roughly 70-30 frequency. So that's another kind of permanent line. And the way we studied it more recently was as a kind of perceptual bistability. So like the rabbit duck illusion, right? You look at it and it looks like one thing, looks like something else. That's kind of what's happening here. There's a bioelectrical pattern that can be interpreted in one of two ways, and that's why they're confused. They're sort of bistable and they can fall in either direction. So that's another thing we did in planaria.
Okay, so that's 2017. I'm going to get you to bring us up to 2020 and then to 2024. But first, what is meant? Explain to myself what is meant when you say Levin lab. So there's also Huberman lab. Do biologists, do professors who are in neuroscience or biology get given a lab by the university? Is this standard? Do you share a lab with other people? What's meant by lab? Is it a room? What is it?
Yeah. Okay. So the way this works is basically that when you're finishing up your postdoc, you do what people call going out on the market, which means you interview at a bunch of places and see who will hire you as a brand new junior faculty member. So when you get a job, and so this is considered your first real independent job because you are now in charge of all your successes and failures. It's all up to you. And typically, yes, at that point you get a lab. So one of the things you do is you negotiate the amount of space you have. Typically, you start off pretty small, and then over time, if you bring in new grants, then you ask for more space and the lab grows. And when they say Levin lab or Huberman lab, what they're just referring to is the all of the research that is controlled by you, where you make the decisions, where that particular person makes the decisions. So physically, for example, I have 3 different locations on this campus where my research is done, and that's just because there isn't one contiguous space. I would be happier to have everything under one, in one place, but they're large spaces, and so there's a few specific locations. All of them are considered part of the Levin lab because, as the principal investigator, I'm the one whose job it is to bring in the external funding to support all the people that work there and to pay for the reagents, and also I'm the PI of these labs. It's their job to be responsible for the good and the bad of what they do. So that's what it means. It just means you're the principal investigator responsible for determining what research happens and what happens to that research. Who does it? You're hiring. You're recruiting people. You're writing grants. That's what it means.
And for the people who are watching, for the people who are listening, there are shots on screen right now of the Levin lab, provided we're able to go later. Okay, so now bring us to 2020 to 2024.
Yeah. So some of the latest, most exciting things that. By the way, one thing I didn't mention that happened around between 2015 and 2020 or so was also Vaibhav Pai's. He's also a staff scientist at my lab. We were able to show that you can actually use reinforcement of bioelectric patterns to fix birth defects. So in frog, it started. Yeah, this started in frog, and we were able to show that there's a range of birth defects induced by other means that you can actually repair. So complex defects of the brain, face, heart, like these really complicated things, you can actually repair them by very simple changes in the bioelectrical pattern. So I think that's a really important actually story because not only is it a path to birth defects, clinical repair of birth defects, but it actually shows how, again, you can take advantage of this highly modular aspect, and you don't have to tattoo instructions onto every cell for something as complex as a brain or a heart. You can give pretty large scale instructions, and then the system does what it does to fix it. So. Okay, so the next couple of big things after that were, first of all, the discovery of xenobots, and this was Doug Blackiston and my group did all the biology for it, and this was in collaboration with Josh Bongard, who's a computer scientist at UVM, and his PhD student at the time, Sam Kriegman, who did all the computer simulations for the work. So this was the discovery of the xenobots and the idea of using epithelial cells from an early frog embryo to prospective skin cells to let them to liberate them from the rest of the embryo, where they're basically getting a bunch of signals that force them to be this boring skin layer on the outside of the animal, keeping out the bacteria.
So they were going to be a skin cell. They weren't yet.
They were well at the it's hard because skin isn't it's not a real precise term, but it's cells that at the time that we took them off the embryo were already committed to the fate of becoming that kind of outer epithelial covering. They hadn't matured yet, but they already knew they were going down that direction. So So we were able to show that when you liberate them from those cues, they actually take on a different lifestyle, and they become a motile, sort of self-contained little construct that swims around and does some really interesting things, including making copies of itself from loose skin cells that you provide, and so on. Shortly thereafter, a couple of years later, we were able to make anthrobots, which is the same thing but with adult human tracheal cells. And part of that, so that has there's a couple of reasons why that's important. Very simply, some people saw xenobots and they thought, well, amphibians are plastic, embryos are plastic. Maybe not shocking that cells from a frog embryo can reassemble into something else, but basically, this resembles a phenomenon in frog developmental biology known as an animal cap. And one way
An animal cap.
It's called an animal cap. The animal cap is just basically that top layer of skin cells, of prospective ectodermal cells. It's called an animal cap. So some people thought about this as a unique feature of frog developmental biology. And so I wanted to get as far away from frog and embryo as possible because I wanted to show that this was a kind of general and a broader phenomenon. So what's the furthest you can get from embryonic frog, adult human? So Gizem Gomushka in my group, who just defended her PhD about a month ago, so she developed a protocol to take donated tracheal epithelial cells from human patients, often elderly patients, and let them assemble into a similar kind of thing, a self-motile little construct that swims around on its own. And one of the most amazing things that these anthrobots do, and this is just the first thing we tried, so I'm guessing there's 100 other things that they can do, but one thing that they can do is if you put them on a neural wound, so in a petri dish you can grow a bunch of human neurons, you take a scalpel, you put a scratch down through that lawn of neurons, so there's neurons here, there's a big wound in the middle. When the anthrobots come in, they can settle down into a kind of we call it a superbot, which is a collection of anthrobots, and 4 days later, if you lift them up, what you see is what they do is they take the two sides of the neural wound and they knit them together, so they literally repaired across that gap. So you can sort of start imagining, there's a couple of things. On a practical level, you can imagine in the future personalized interventions where your own cells, no heterologous, synthetic cells, no gene therapy, but your own cells are behaviorally reprogrammed to go around your body and fix things in the form of these anthrobots. You don't need immune suppression because they're your own cells, right? And so you can imagine those kinds of interventions. It's interesting because a lot of the interventions we use now, we use drugs and we use materials, screws and bolts and things, and then occasionally we use like a pacemaker or something. But generally, our interventions are very low agency, and ultimately we'll have like smart implants that make decisions for you. But mostly they're very low agency kinds of things. And when you're using low agency tools, the kinds of things you can do are typically very brittle. In other words, this is why it's hard to discover drugs that work for all patients. You get side effects, you get very differential efficacy in different patients because you're trying to micromanage at the chemical level a very complex system.
You don't just want agency, you want agency that's attuned to yourself, because you get someone else's agency. It's not great for you.
Exactly, which is why your own cells coming from your own body, they share with you all the priors about health, disease, stress, cancer. They're part of your own body. They already know all of this. And so part of this is creating these, understanding how to create these agential interventions that can have these positive effects. We didn't teach the anthrobots to repair neural tissue. We had no idea. This is something they do on their own. Like, who would have ever thought that your tracheal cells, which sit there quietly for decades, if you let them have a little life of their own, they can actually go around and fix neural wounds, right?
Well, you must have had some idea that this would be possible, otherwise you wouldn't have tested it, no?
True, yes. This is true for a lot of stuff in our lab where people say, well, did you know that was going to happen? And so on the one hand, no, because it's a wild and it wasn't predicted by any existing structures. On the other hand, yes, because we did the experiment and that's why I did it, because I had an intuition that that's how this thing would work. So did I know that it was going to specifically repair peripheral innervation? No, but I did think that it would that among its behavioral repertoire would be to exert positive influence on human cells around it. And so this was a convenient assay to try. We have 100 more that we're going to try. There's all kinds of other stuff.
Yes, I see. So you're testing out a variety.
Correct. You have to start somewhere, right? And so we said, okay, so Gizem and I said, well, why don't we try some a nice easy neural scar? There's many other things to try. So that's kind of the practical application, but the kind of bigger intellectual issue is, much like with the xenobots, what's cool about making these sorts of synthetic constructs is that they don't have a long evolutionary history in that form factor. There's never been any xenobots. There's never been any anthrobots in the evolutionary history. The anthrobots don't look anything like a human stages of human development. And so the question arises, where do their form and behavior come from then, right? And so this is where you get back to this issue of the Platonic space, right? If you can't pin it on eons of specific selection for specific functions, where do these novel capabilities come from? And so I really view all of these synthetic constructs as exploration vehicles. They're ways to look around in that Platonic space and see what else is out there. We know normal development shows us one point in that space that says this is the form that's there. But once you start making these synthetic things, you widen that your view of that latent space as to what's actually possible. And I see this research program as really investigating the structure of that Platonic space in the way that mathematicians, people make the map of mathematics, right? And there's a sort of structure of how the different pieces of math fit together. I think that's actually what we're doing here when we make these synthetic things, is we're making vehicles to observe what else is possible in that space that evolution has not shown us yet. Yeah. So, and then you can do interesting things like, and this is still unpublished, but you can ask questions like, what do their transcriptomes look like? What genes do xenobots and anthrobots express? And without blowing any of the sort of surprise, the paper should be out sometime this year. massively new transcriptional profiles in these things. No drugs, no synthetic biology circuits, no genomic editing. Just by virtue of having a new lifestyle, they adapt their transcriptional profile. The genes that they express are quite different. So that'll be an interesting study. And then, for the rest of it, I mean, what we've been doing in the last few years is trying to bring a lot of the work that we've done earlier into clinically relevant models. So the cancer stuff has moved from frog into human cells and organoids, spheroids. So human cancer spheroids and glioblastoma, colon cancer, stuff like that. The regeneration work has moved from frog into mice. So, and this is it's coming along. I'm not claiming any particular result yet. This is I should also say there's an invention. What do you call it? A disclosure here I have to do because we have a couple of companies now, and so I have to do a disclosure that in the case of regeneration, so Morphoceuticals is a company that Dave Kaplan and I have. So David is a bioengineer here in at Tufts, and he and I have this company aiming at limb regeneration and more broadly bioelectrics in regeneration. So the cancer, the limb regeneration stuff. More experiments in trying to understand how to read and interpret the information that flows across levels. So we know cells exchange electrical signals to know how to make an embryo. Turns out embryos actually communicate with each other. So that's been a really exciting finding recently for Angela Tung in my group, which just got her PhD as well, where we studied this embryo to embryo communication, showing that groups of embryos actually are much better at resisting certain defects than individuals, and they have their own transcriptional profile. So I call it a hyperembryo because it's like the next level. They have an expression, a transcriptome that is different from normal embryos developing alone, let's say. So that's pretty exciting. Those are the kinds of things we've been focused on.
Okay, now we're going to end on advice for a newcomer to biology. They're entering the field. What do you say?
Boy, step one is to ignore most people's advice. So that's I don't know how helpful that will be, but I actually have I have a whole thing about, maybe we can put up a link. I have a whole long description of this on my blog. So on my blog, I have a thing that basically talks about advice.
Okay, that's on screen. Also, you should know that the previous research by Angela Tung and Gamaskaya.
Yeah, Gizem Gamaskaya.
We did a podcast together, so that link will be on screen as well. There's also another podcast with Michael Levin, which is on screen, and then another one which is on screen with Chris Field and Carl Friston. Another one with Michael Levin and Yosha Bach. That's on screen. So Michael is a legend on theories of everything. Okay, so does your advice for the biologist differ from your advice to the general scientist entering the field?
I mean, the most important thing I'll say is I do not in any way feel like I could be giving anybody advice. I think that there are so many individual circumstances that I'm not going to claim I have any sort of.
How about what you would have wished you had known when you were 20?
Yeah. So this is pretty much the only thing I can say about any of this. That even very smart, successful people are only well calibrated on their own field, their own things that they are passionate about. They're not well calibrated on your stuff. So what that means is that if somebody gives you, so this is all about, it's kind of meta advice. It's all about advice on advice. And the idea is that when somebody gives you a critique of a specific product, so let's say you gave a talk or wrote a paper or you did an experiment, somebody's critiquing what you did, right? That's gold. So squeeze the hell out of that for any way to improve your craft. What could I have done better? What could I have done better to do a better experiment? What could I have done better in my presentation so that they would understand what I want them to understand? That's gold. The part where everybody gives large scale advice, work on this, don't work on that, focus, don't think of it this way, think of it that way. All of that stuff is, generally speaking, better off ignored completely. So people are really not calibrated on you, your dreams in the field, your ideas. It does not pay to listen to anybody else about what you should be doing and how. You really need to be, everybody needs to be developing their own intuition about what that is and testing it out by doing things and seeing how they land. And I think that most everything that we've done along the way that's interesting, and certainly we've had plenty of dead ends and made plenty of mistakes, but most of the interesting things that our lab has done along the way, very good, very successful, smart people said, don't do this. There's no way this is going to lead to anything. And so the only thing I know is that paths in science are nobody has a crystal ball. Paths in science are very hard to predict, and people should really be very circumspect about taking extremely seriously specific critiques of specific things that will help them improve their process versus these large scale, sort of career level things that I don't think you should be taking almost anybody's advice about that.
Can you be specific and give an example of where you liked the minutia of a critique and then where you disliked the grand scale critique?
Yeah, I mean, the minutia, it happens every day because every day we get comments on let's say a paper submission and we see, and somebody says, well, it would have been better if you included this control, or I don't get it because so, it's clear that the reviewer didn't understand what you were trying to get at, and so that's on us. That's on us to describe it better, to do a better experiment that forces them to accept the conclusion, whether they like it or not. Right? The best experiment is one that really it forces the reader to a specific conclusion, whether or not they wanted to go there. It's irresistible. It's so compelling. It's clean. It's compelling. So that kind of stuff happens on a daily basis, where you see what somebody wasn't able to absorb from what you did, and you say, okay, how can I do this experiment better? What kind of a result would have gotten us to a better conclusion where everybody would have been able to see? So that stuff happens all the time. the other kind of thing, I mean, I'll give you an example from the tail regeneration kind of era. We showed that normally when tadpoles normally regenerate their tail, there's a particular proton pump that's required for that to happen. You know, a proton pump in the frog embryo. And so what we showed is that you can get rid of that proton pump, and then the tail stops regenerating, and then you can rescue it by putting in a proton pump from yeast that has no sequence of structural homology to the one you knocked out of the frog, but it has the same bioelectric effect, right? And that's how you show that it really is bioelectricity, right? So we got 2 reviews on that paper, and the first reviewer said, oh, you found the gene for tail regeneration. That proton pump is the gene for tail regeneration. Get rid of all the electrical stuff. You don't need it. You found the gene for tail regeneration. The second reviewer said, oh, the gene obviously doesn't matter because you just replace it by the proton pump. So by the one from yeast, yeah, get rid of all of that and just do the bioelectrical stuff, right? So that shows you right away that 2 different perspectives, right? Each person had a particular way they wanted to look at it. They had exactly opposite suggestions for what to throw out of the paper, and only together do those 2 perspectives explain that what's going on here is that yes, the embryo naturally has a way of producing that bioelectrical state, but what actually matters is not the gene. It's not how you got there. It's the state itself, right? And so that kind of thing. Those kind of perspectives, or you know the people who are upset at you know, for example, calling xenobots bots, right? We call them bots because we think it's a biorobotics platform. So one thing that happens is that you've got the people that are sort of from the organicist tradition, and they'll say it's not a robot, it's a living thing. How dare you call it a robot? And part of the issue is that all of these terms, much like the cognitive terms that we talked about, it's not that the xenobot is or isn't a robot. It's simply the idea that by using these different terms, what you're signaling is what are some of the ways that you could have a relationship with it. So, for example, we think that we might be able to program it to use it for useful purposes. That's what the terminology bot emphasizes. Do I think it's only a xenobot? Absolutely not. I also think it's a proto-organism with its own its own limited agency and its own things that we haven't published yet, which we're working on. Its their learning capacity and so on. So you often run into that, is that people think everything should only be one thing, and that this is all a debate about which thing is it. And I don't think that's true at all. There's another, just kind of one last example. Again, having to do with terminology. Somebody said to me once, people are very resistant to the use of the word memory for some of the things that we study. And she said, why don't you just come up with a new term, schmemory or something, and then nobody has to be mad. You can say, okay, like this is human learning is memory, and then this other thing where these other things learn, well, that's memory. So then. And that's the kind of
intelligence.
Yeah.
Why are you calling it intelligence?
Yeah, exactly. And that's the kind of, okay, so that's just an example of the kind of advice you might get from somebody. And in a certain sense, it's true that if you do that, you will have fewer fights with people who are very purist about, they want memory to be in a very particular box. You'll have less fights with those. Those are true. But bigger picture though, imagine, so there's Isaac Newton and the apples fall. I mean, I know it didn't really probably happen, but let's, the apples fall in the tree. And he says, okay, so gravity, I'm going to call gravity the thing that keeps the moon, in orbit of the Earth. And then I'm going to call schmavity the thing that makes the apple fall. That way there won't be any arguments, right? Like, yeah, but what you've done there is you've missed the biggest opportunity of the whole thing, which is the unification. The thing that the fact that actually, no, the hill you want to die on is that it really is the same thing. You don't want a new term for it. So that's just an example of the kind of, it's good advice if you want to avoid arguments, but if your point is that no, it actually is, that's the whole point. We need to have a better understanding of memory. This is I want those arguments. Then that's something else. And that's the kind of strategic thing that you should decide on your own what you want to do.
Now, in that case, why couldn't you just say, actually, it wouldn't have been a mistake for Newton to call this one gravity 1 and that one gravity 2 until he proves that they're the same mathematically, just like they're different. There's inertial mass, and then there's another form of mass, and then you have the equivalence principle.
Yeah, you could. The thing is that my point is
the issue is that we never know a priori whether we're supposed to unify or distinguish.
That's correct. Yes. No, true. A priori, you don't know. And so the question is, in your own research program, which road do you want to go down to? Because if you commit to the fact that that they're separate, you don't try the unification, right? You try the unification. You spend your time. I mean, it takes years, right? You spend time and effort in a particular direction because you feel it will pay off. If it were truly, it could be this or that. Are you going to spend 10 years on one of these paths, right? You really need to in science. There's no do-overs. Like you commit those 10 years are gone. So you need to have a feeling. You need to have an intuition of which way it's going to go. And you definitely don't need to declare ahead of time that I know how it's going to turn out, because you don't. But you do need to know, despite everybody telling me this or that, I am going to commit. That's really all you have, right? You don't have any kind of crystal ball. You don't have a monopoly on the truth. But what you do have is a responsibility to manage the limited time that you have. So how are you going to spend your 10 years? And it's going to be hard, right? Lots of blood, sweat, and tears. It's a hard job. There's constant criticism. And that's how science goes. Lots of stress. But so now the question is, are you going to have that stress following somebody else's research agenda or yours? You'll still be old and stressed out by the end of it. But the question is, will you have tested out your own best ideas or somebody else's view of what science should be? That's my only hope.
Well, Michael, speaking of limited time, I appreciate you spending yours with me and with the crew here today.
Super. Thanks.
So there you have a biology's view on archetypes, on forms, on function, and I think it's very exciting what he's doing, and he definitely, I would put him in the Nobel Prize category for what he's doing. And picking up on what Darcy Wentworth Thompson did 100 years ago, but really with far greater resources. He's using computers. He's got access to a ton of money. You know, none of this was true for Darcy Wentworth Thompson. He was who he was, you know.
Oh,
So
but he's building on
yeah, he's building
Darcy's
on
work. Yeah.
all kinds of things. So, but he's also, as you can see, he's asking fundamental questions, and typically what he's saying is, I'm betting on my intuition that if we ask this question, this will take us in a really great direction, and I'm not going to spend my time looking over what someone else did when I'll miss the opportunity to go do some really new unifying thing that just never came up. So
I love the way he works with things in these modular terms, you know, like exporting these cells, and then what you're discovering is that it's this ability and not that ability that can be exported and used in this completely new context. And so he just sort of sees things in terms of its application or its potential.
well, that's what he said. He said, I don't want to just do theory. I want to actually have some impact in the world I live in now. And but it's really the fact that he's doing lots of experiments.
Right.
Okay, a lot of these other people just do thought experiments. He's doing real experiments with all these. He's getting these really bright kids that are just working. You know, they live on coffee and they work 20-hour days for years on end, and they do these amazing things. So, you know,
Right.
I met, for example, at MIT, there was a group that did the Bose-Einstein condensate, which is this form of matter that uh, Bose, who was an Indian, proposed and Einstein picked up on, but it was done theoretically, and no one knew, could you actually do one of these condensates, in which they did it, you know, close to absolute zero. And when I talked to the professor and then I talked to the graduate student, it's obvious the graduate student did 99% of the work. Sitting there, you know, getting vacuum pumps to work and writing software and, you know, getting the lasers to all work that were driving all this thing. I mean, that's what he's leveraging off of, and they're going down lots of blind alleys, but some of them are going to pay off. The problem he said is, I just don't know which ones. So
Right. Or in what way. You don't always know what
no,
it's actually
or
showing.
or you'll look at it and you'll totally miss the
Right.
real payoff. Whatever it is, it'll be someone else that looks at it and says, "Oh, well, you could do this."
Right.
What? So,
Well, this
but
could,
it's all, again, just to pick up, it's this notion of these forms that drive everything, these algorithms that are above it all and shape these things. And it's a very different way of looking at biology.
Right.
So,
I like the way it shows the complexity of the unity. That's something that always intrigues me,
oh yeah.
that unity doesn't have to be uniform, you know, across the board to be uh, unified.
So I think next time we'll go back to Jung and look at archetypes, because there's a lot of new videos out there on Jung and what people have discovered that was buried in his writings and stuff, and there'll be more than enough there. I think we have enough stuff to carry us all the way through the summer from looking at what in the world is out there on the web. So the question is, will the world end before we get there?
Right.
That's the other question. So
Or at least the United States.
yeah.
Well,
Yeah.
what did this trigger for you, Bruce, as a Buddhist? What did this weekly episode?
Well, it's certainly a different view of the mind. One of the perspectives I have is that when you start meditating and start understanding the different levels of the mind, And well, the big one is that for when you start transcending the conditioned mind, and you start finding the levels of the reality that have been there forever, it gives you a very different perspective. Because then you understand, well, in Buddhism, they call it natural mind. This is the mind that's been there forever and is understands levels of reality that the conditioned mind can't even go to at all. That's my main perspective. Because when you start meditating and you start transcending some of the normal formations of the mind, it changes everything. You understand things that you've never seen before at all.
Yeah, you remember he was asked, well, what do you mean about this being higher or lower? And I was thinking, he doesn't, it doesn't sound like he does a lot of meditation. He might,
No, that's right. That's right.
you might, but I don't think he's picked up on this as an idea. And also the as he said, that the notion of consciousness is you're an integral part of it. So how do you sort this out? What experiments are you going to run? Because you're in the middle of it. Okay, it's you. So now what?
Yeah, right. I mean, basically, he's on concepts and understanding theories. Whereas when you start to meditate, you start seeing these things directly with your own mind. You have awareness of things that you've never could have imagined were possible at all. And they start becoming what is actually normal understanding of reality. It's just a whole different perspective of exploring consciousness.
Yeah, and that's, who knows? Maybe he'll get a couple of Buddhist, postdocs. Who knows?
Well, Ron was a good example because he showed you what meditation where it can go.
Yeah. Ron is a great example. It just, he's got to
need 10 more.
Yeah. Yeah. No, you could do worse. So, well, another week. We did it.
Okay.
We will see you next week. Have a good.
Okay. Yeah.
Bye-bye.
Happy July.
Bye.